Practical activities, skills, and interrelationships in the syllabus. In this section, we will delve into the practical activities, skills, and interrelationships covered in the provided syllabus. This will involve providing a detailed explanation and exploration of the various components and their relevance within the context of the syllabus. 1. Practical activities. Practical activities refer to hands-on exercises, experiments, or tasks that are designed to enhance the application of theoretical knowledge. These activities often involve real-world scenarios or simulations that allow students to actively engage with the subject matter. Practical activities provide a valuable opportunity for students to develop practical skills, critical thinking abilities, and problem-solving techniques. They also encourage active participation, collaboration, and experimentation. 2. Skills. Skills play a crucial role in the syllabus as they are the abilities that students acquire or improve upon throughout the course. These can include subject-specific skills, such as laboratory techniques in a science course, as well as transferable skills like communication, research, and analytical thinking. The syllabus aims to cultivate a diverse range of skills to equip students with the necessary tools to succeed both academically and in their future endeavors. 3. Interrelationships. Interrelationships refer to the connections and interactions between different topics or subject areas within the syllabus. These connections are essential in promoting a holistic understanding of the subject matter and fostering interdisciplinary thinking. By exploring the interrelationships, students can recognize the links between various concepts, draw parallels, and identify overarching themes. This approach encourages a deeper comprehension of the material and helps students make connections across different areas of knowledge. By incorporating practical activities, developing skills, and emphasizing interrelationships, the syllabus aims to provide a comprehensive and enriching learning experience. This approach encourages students to actively engage with the content, apply their knowledge in practical settings, and develop a broader understanding of the subject matter. Cells, the building blocks of life. In this section, we will explore the fascinating world of cells, which are the fundamental units of life. Cells play a crucial role in all living organisms and are responsible for carrying out essential functions necessary for survival. By providing a detailed explanation and expanding on the topic, we can gain a deeper understanding of the significance and complexity of cells. 1. Cells as basic units. Cells are the basic structural and functional units of all living organisms. They are incredibly diverse in their forms and functions but share some common characteristics. Cells are microscopic in size and can only be observed using powerful microscopes. Despite their small size, cells are highly organized and contain various components that enable them to carry out specific tasks. 2. Cell structure and components. Cells consist of different structures and organelles, each with a specific function. The two primary types of cells are prokaryotic and eukaryotic cells. Prokaryotic cells, such as bacteria, lack a nucleus and other membrane-bound organelles. Eukaryotic cells, found in plants, animals, fungi, and protists, have a distinct nucleus and various membrane-bound organelles, including mitochondria, endoplasmic reticulum, Golgi apparatus, and more. 3. Cell functions. Cells perform a wide range of functions necessary for the survival and functioning of organisms. These functions include energy production, protein synthesis, nutrient uptake, waste elimination, reproduction, and cell signaling. Each cell type has specific functions depending on its specialization within the organism. For example, nerve cells transmit electrical signals, muscle cells enable movement, and red blood cells transport oxygen. 4. Cell division and reproduction. Cells have the ability to divide and reproduce, allowing organisms to grow, develop, and repair damaged tissues. The two primary methods of cell division are mitosis and meiosis. Mitosis is a process by which cells replicate themselves, resulting in two identical daughter cells. Meiosis, on the other hand, occurs in specialized cells called germ cells and leads to the formation of gametes, sperm and egg cells, with half the genetic information of the parent cell. Understanding the intricacies of cells is crucial in many scientific disciplines, including biology, genetics, medicine, and bioengineering. By studying cells, scientists can gain insights into the functioning of living organisms and develop a deeper understanding of diseases and their treatments. Cells are truly remarkable entities that form the foundation of life as we know it. Exploring cells and cell structures through electron micrographs, DR. In this section, we will delve into the fascinating world of cells and their structures by utilizing electron micrographs. Electron micrographs provide high-resolution images that allow for detailed examination and labeling of various cell components. This approach offers a deeper understanding of cellular organization and function, providing valuable insights into the microscopic world. 1. Electron micrographs, a window into the microscopic realm. Electron micrographs are images captured using an electron microscope, a powerful tool that uses a beam of electrons to visualize objects at extremely high magnification. Compared to traditional light microscopy, electron microscopy provides superior resolution, enabling us to observe cellular structures with intricate detail. By examining electron micrographs, we can gain a clearer picture of cells and their structures. 2. Drawing and labeling cells. When drawing cells based on electron micrographs, it is essential to accurately represent the observed structures and label them appropriately. These structures may include the cell membrane, nucleus, cytoplasm, organelles, and other cellular components. Careful observation and understanding of the micrograph will guide the drawing process, ensuring accurate representation of the cell's morphology. 3. Cell structures and labeling. Electron micrographs allow us to identify and label various cell structures and organelles. For instance, within a eukaryotic cell, we can observe the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and more. Each structure has distinct characteristics and functions, contributing to the overall functioning of the cell. Proper labeling ensures clarity and facilitates the understanding of cellular organization. By utilizing electron micrographs to draw and label cells, we can visualize cellular structures with remarkable detail. This approach not only enhances our comprehension of cells but also provides a visual representation that aids in communication and further analysis. Electron micrographs are invaluable tools in the field of cell biology, enabling scientists and researchers to explore the intricate architecture of cells and unravel the mysteries of their inner workings. Exploring the microscopic world, examining cells under a microscope, OR. In this section, we will embark on a journey into the microscopic world by examining a diverse range of cells under a microscope. By carefully observing cells through a microscope, we can uncover their intricate structures, characteristics, and functions. This approach enables us to gain valuable insights into the diversity of cells and the complexity of life at the cellular level. 1. Microscopic examination, revealing the hidden world. Microscopic examination involves the use of a microscope to magnify and visualize cells that are otherwise invisible to the naked eye. By employing different types of microscopes, such as light microscopes or electron microscopes, we can explore cells at varying levels of detail and resolution. This allows us to observe and analyze their structural features and functional properties. 2. Cell types and diversity. Cells exhibit tremendous diversity in terms of their size, shape, and organization. By examining different cell types, we can appreciate the remarkable variations that exist across organisms. These cell types may include animal cells, plant cells, fungal cells, bacterial cells, and more. Each cell type possesses unique characteristics and adaptations that enable it to fulfill specific functions within the organism. 3. Cellular structures and features. Microscopic examination of cells allows us to identify and study various cellular structures and features. These structures include the cell membrane, nucleus, cytoplasm, organelles, such as mitochondria, endoplasmic reticulum, Golgi apparatus, and specialized cellular components. By observing these structures, we can gain insights into their morphology, organization, and their roles in cellular processes. 4. Functional insights. Through microscopic examination, we can also infer the functions and activities of cells. For example, by observing the presence of chloroplasts in plant cells, we can deduce their role in photosynthesis. Similarly, the presence of cilia or flagella in certain cells indicates their involvement in cellular movement. Microscopic examination allows us to link cellular structures to their respective functions and understand how cells contribute to the overall functioning of organisms. By exploring a variety of cells under a microscope, we unravel the intricacies of cellular life. This process enhances our understanding of the diverse forms and functions of cells, providing a foundation for advancements in biology, medicine, and other scientific disciplines. Microscopic examination serves as a gateway to the microscopic world, revealing the wonders and complexities of life at the cellular level. Creating cell models, utilizing everyday materials for construction, PD/DR. In this section, we will explore the exciting process of constructing cell models using materials readily available in both home and laboratory settings. Building cell models provides a hands-on approach to understanding the intricate structures and components that make up cells. By utilizing various materials creatively, we can recreate the complexity of cells in a tangible and visually appealing manner. 1. Constructing cell models, a visual and tactile learning tool. Cell models serve as visual representations that aid in comprehending the organization and functionality of cells. By constructing models, we engage in a practical and immersive learning experience that allows us to explore the intricate details of cellular structures. These models can be used in educational settings, research projects, or as visual aids for presentations. 2. Materials selection. Constructing cell models requires selecting appropriate materials that resemble the various components of cells. Common household items such as clay, Play-Doh, styrofoam, cardboard, pipe cleaners, buttons, and craft supplies can be used creatively to represent different cellular structures. In a laboratory setting, more specialized materials like plastic or foam balls, colored markers, and wires may also be available for constructing detailed and accurate models. 3. Cell structure representation. When building cell models, it is essential to consider the accurate representation of different cellular structures. For example, the cell membrane can be represented by a thin layer of clay or Play-Doh surrounding the model. Nucleus, mitochondria, endoplasmic reticulum, and other organelles can be constructed using different materials and shapes to resemble their unique characteristics. Labeling the structures further enhances the educational value of the model. 4. Model customization and detailing. Creating cell models allows for customization and detailing to highlight specific features or adaptations of cells. For instance, in plant cell models, the addition of chloroplasts made from green clay or small green beads emphasizes their role in photosynthesis. Similarly, adding cilia or flagella made from pipe cleaners or thin wires to certain cell models can illustrate cellular movement. Constructing cell models using accessible materials fosters creativity, critical thinking, and a deeper understanding of cell structures and functions. Whether in a classroom or laboratory, these models serve as effective educational tools, enhancing engagement and knowledge retention. By engaging in the construction process, students and researchers gain a hands-on appreciation for the complexity and beauty of cells, bringing the microscopic world to life. Skills, equipping students for success. In this section, we will explore the importance of skills and their role in preparing students for success. Skills are not only essential for academic achievement but also for personal growth, career development, and adaptability in a rapidly changing world. By providing a detailed explanation, we can understand the significance of skills and how they contribute to overall competence and lifelong learning. 1. Acquisition and development of skills. Skills are abilities that individuals acquire and develop through learning, practice, and experience. They encompass a wide range of competences, including both technical or domain-specific skills and transferable skills that can be applied across different contexts. Acquiring skills requires active engagement, perseverance, and a willingness to learn and adapt. 2. Importance of transferable skills. Transferable skills, also known as soft skills or employability skills, are highly valuable in various aspects of life. These skills include communication, critical thinking, problem-solving, teamwork, adaptability, creativity, and leadership. Transferable skills enable individuals to navigate complex situations, collaborate effectively, think critically, and communicate their ideas clearly. These skills are often sought after by employers as they contribute to professional success and career advancement. 3. Skill development in education. Education plays a vital role in skill development by providing opportunities for students to acquire and enhance their skills. Educational institutions strive to incorporate skill development within the curriculum, through project-based learning, group activities, presentations, research projects, and practical experiences. By integrating skill-building activities, students can develop a well-rounded set of competencies that prepare them for future challenges. 4. Lifelong learning and skill adaptation. Skills are not static, they need to be continuously nurtured and adapted to keep pace with changing circumstances and advancements. Lifelong learning is essential to stay relevant and thrive in an evolving world. The ability to learn new skills, adapt to technological advancements, and embrace continuous improvement is crucial for personal and professional growth. 5. Skill application in real-world contexts. The true value of skills lies in their application in real-world contexts. Whether it is problem-solving in a work environment, effectively communicating ideas, or collaborating with diverse teams, skills enable individuals to navigate challenges, seize opportunities, and achieve their goals. The practical application of skills enhances competence and contributes to success in various endeavors. By recognizing the importance of skills, educational institutions and individuals can prioritize skill development alongside academic knowledge. The acquisition and refinement of transferable skills empower individuals to navigate complexities, adapt to change, and excel in their chosen paths. Skills are not only crucial for immediate success but also for lifelong learning and continuous personal and professional development. Drawing. Enhancing accurate representation of cell structures and organelles, DR. In this section, we will explore the skill of drawing and its significance in accurately representing the structures and organelles of cells. Drawing is a valuable skill that aids in visualizing and communicating scientific concepts effectively. By providing a detailed explanation, we can understand how drawing skills contribute to the accurate depiction and comprehension of cell structures. 1. Drawing as a visual communication tool. Drawing serves as a powerful visual communication tool, allowing students to express their understanding of complex concepts. When it comes to cell structures and organelles, drawing enables students to depict these intricate components with clarity and precision. Through drawing, students can convey their observations, interpretations, and conceptualizations of cell structures to others. 2. Enhancing observation and attention to detail. The skill of drawing requires students to carefully observe and analyze the structures and organelles of cells. By focusing on details such as shape, size, and arrangement, students develop a deeper understanding of the subject matter. Drawing encourages attention to fine details, which is crucial for accurately representing cell structures and capturing their specific characteristics. 3. Developing spatial awareness. Drawing cell structures helps students develop spatial awareness, enabling them to depict the three-dimensional nature of cells on a two-dimensional surface. Understanding how organelles are positioned within the cell and their relative sizes and distances enhances the accuracy of the drawings. Through practice, students hone their ability to represent spatial relationships effectively. 4. Integration of knowledge and visualization. Drawing requires students to integrate their knowledge of cell structures with their visualization skills. By combining their understanding of cell biology concepts with their ability to depict those concepts visually, students create drawings that effectively communicate the features and functions of cell structures and organelles. This integration fosters a deeper comprehension and retention of the subject matter. 5. Promoting creativity and critical thinking. Drawing encourages creativity and critical thinking as students explore different ways to represent cell structures and organelles. It allows for the application of artistic techniques, such as shading, highlighting, and perspective, to enhance the visual impact of the drawings. Through creative expression, students develop their own unique style while engaging in analytical thinking to accurately represent cellular components. The skill of drawing plays a significant role in accurately representing the structures and organelles of cells. It promotes observation, attention to detail, spatial awareness, and the integration of knowledge and visualization. By honing their drawing skills, students can effectively communicate their understanding of cell biology and contribute to the scientific community's visual representations of cellular structures. Observation and recording, facilitating detailed cell analysis and documentation, ORR. In this section, we will explore the importance of the skill of observation and recording in enabling students to make detailed observations of cells and effectively document their findings. This skill is crucial in scientific investigations, as it enhances accuracy, attention to detail, and the ability to analyze and communicate observations. By providing a detailed explanation, we can understand how observation and recording skills contribute to the comprehensive study of cells. 1. The role of observation in cell analysis. Observation is the foundation of scientific inquiry, and it plays a central role in studying cells. Through careful observation, students can examine cell structures, their characteristics, and behaviors. By keenly observing cells under a microscope or through other means, students can identify and document key features, such as cell shape, size, color, and the presence of organelles. Observation provides the necessary information for further analysis and understanding of cell biology. 2. Attention to detail and accurate recording. Developing the skill of observation involves cultivating attention to detail. Students must pay close attention to the subtle variations and characteristics of cells to capture accurate information. Accurate recording of observations involves noting down important details, measurements, and any other relevant data. This meticulous approach ensures that recorded findings are reliable and can be referenced for future analysis or comparison. 3. Documenting findings for analysis and communication. Recording observations allows students to document their findings in a structured and organized manner. This documentation serves as a valuable resource for analysis, comparison, and future reference. Students can refer back to their recorded observations to identify patterns, make connections, and draw conclusions about cell structures and functions. Furthermore, documented findings can be effectively communicated to others, such as peers or instructors, through written reports or visual representations. 4. Enhancing analytical skills. The skill of observation and recording enhances students' analytical abilities. By consistently observing cells and documenting their findings, students develop a systematic approach to analyzing complex biological structures. They learn to identify patterns, draw meaningful conclusions, and make connections between observed phenomena and underlying scientific principles. These analytical skills foster critical thinking and a deeper understanding of cell biology. 5. Developing a scientific mindset. Cultivating the skill of observation and recording nurtures a scientific mindset in students. It encourages curiosity, objectivity, and a desire for evidence-based understanding. Through systematic observation and accurate recording, students learn to approach scientific investigations with rigor, discipline, and a commitment to detail-oriented work. This mindset prepares them for future scientific endeavors and fosters a lifelong appreciation for the scientific method. Observation and recording skills are essential in the study of cells, enabling students to make detailed observations, accurately document their findings, and analyze cellular structures and functions. These skills facilitate scientific inquiry, enhance analytical thinking, and contribute to the advancement of cell biology knowledge. By honing their observation and recording skills, students become adept at capturing and analyzing the intricacies of the microscopic world, unlocking deeper insights into the fascinating realm of cells. Structure and function relationships, understanding the interplay of cell structures and functions. In this section, we will delve into the concept of structure and function relationships in cells, exploring the intricate interplay between cellular structures and their specific functions. By providing a detailed explanation, we can deepen our understanding of how the unique features and organization of cell structures directly contribute to their functional roles. 1. Structural organization and cellular functions. The structure of a cell is intricately linked to its specific functions. Each cellular structure, such as the cell membrane, nucleus, mitochondria, endoplasmic reticulum, or Golgi apparatus, possesses distinct characteristics and arrangements that enable it to perform specific tasks. Understanding how the structure of each component contributes to its function allows us to grasp the remarkable efficiency and complexity of cellular processes. 2. Specialization and functional adaptations. Cells exhibit specialization, where specific structures are modified or developed to carry out specialized functions. For example, in plant cells, chloroplasts are specialized structures responsible for photosynthesis, utilizing their unique structure that contains chlorophyll to capture light energy. Similarly, in nerve cells, the elongated shape and branching structure of dendrites facilitate the transmission of electrical signals. 3. Complementary structure-function relationships. The relationship between cell structure and function is often complementary, with structures evolving to fulfill specific functions optimally. This relationship ensures that cells are equipped with the necessary tools to carry out their designated roles efficiently. For instance, the extensive network of membranes and organelles within a eukaryotic cell allows for compartmentalization and efficient synthesis, storage, and transport of molecules. 4. Adaptations for environmental demands. Cells can adapt their structures to meet the demands of their environment. Environmental factors, such as temperature, pH, or the availability of nutrients, can influence the organization and functioning of cell structures. For example, in cells exposed to high levels of stress, the endoplasmic reticulum may undergo changes to increase its capacity for protein folding and quality control. Understanding the relationships between cell structures and their functions is crucial for comprehending how cells maintain homeostasis, carry out specialized tasks, and respond to environmental cues. This knowledge is fundamental in fields such as cell biology, physiology, and medicine. By studying these relationships, students can appreciate the remarkable design and functionality of cells, laying the groundwork for further exploration and discovery in the biological sciences. Nutrition, fueling cells for growth and function. In this section, we will delve into the topic of nutrition, exploring its crucial role in providing cells with the necessary fuel for growth, maintenance, and optimal functioning. By providing a detailed explanation, we can understand the significance of nutrition and how it directly impacts cellular processes. 1. The importance of nutrition for cells. Nutrition plays a vital role in sustaining the life and functionality of cells. Cells require various nutrients, including macronutrients, such as carbohydrates, proteins, and fats, and micronutrients, such as vitamins and minerals, to carry out essential processes. These nutrients provide energy, building blocks, and cofactors that support cellular growth, repair, and the synthesis of biomolecules. 2. Energy production. Nutrition serves as a primary source of energy for cells. Through processes like cellular respiration, cells convert nutrients, particularly carbohydrates and fats, into adenosine triphosphate, ATP, a molecule that stores and releases energy. ATP fuels the diverse activities within cells, including metabolic reactions, movement, and the transport of molecules across membranes. 3. Building blocks for cellular components. Nutrients provide the necessary building blocks for synthesizing cellular components. For example, proteins are made up of amino acids obtained from dietary protein sources. These amino acids are essential for protein synthesis, which is crucial for cellular growth, repair, and the functioning of enzymes, receptors, and structural proteins. Similarly, lipids obtained from dietary sources serve as components of cell membranes and play a role in signaling and energy storage. 4. Micronutrients and cellular functions. Micronutrients, including vitamins and minerals, are essential for maintaining cellular functions. They act as cofactors in enzymatic reactions, facilitating metabolic processes and cellular signaling. For instance, vitamins like vitamin C, vitamin D, or vitamin B12 play critical roles in supporting immune function, bone health, and energy metabolism. Minerals like calcium, iron, and zinc are necessary for various cellular processes, including muscle contraction, oxygen transport, and DNA synthesis. 5. Cellular nutrition and health. Adequate nutrition is crucial for overall cellular health and the prevention of diseases. Imbalances or deficiencies in nutrients can disrupt cellular processes, impair growth, and lead to various health issues. For example, insufficient intake of essential nutrients like vitamin C can result in weakened immune function and increased susceptibility to infections. Likewise, excessive consumption of unhealthy fats or sugars can lead to metabolic disorders and contribute to the development of chronic diseases. Understanding the role of nutrition in cellular processes provides insights into the importance of a balanced and nutrient-rich diet. Proper nutrition ensures that cells receive the necessary fuel and building blocks for optimal growth, functioning, and overall health. By maintaining a well-balanced diet, individuals can support cellular health, enhance their well-being, and reduce the risk of nutritional deficiencies and associated diseases. Differentiating food substances, performing tests for starch, proteins, lipids, and reducing sugars, OR, millimeter, AI. In this section, we will explore the process of performing tests to distinguish among various food substances, including starch, proteins, lipids, and reducing sugars. These tests enable us to identify and differentiate the presence of these substances in different food samples. By providing a detailed explanation, we can understand the principles behind each test and their significance in food analysis. 1. Test for starch. The iodine test is commonly used to detect the presence of starch in food samples. Iodine reacts with starch molecules to form a bluish-black color complex. By adding a few drops of iodine solution to a food sample, we can observe the color change, indicating the presence of starch. This test is particularly useful in identifying starchy foods like potatoes, rice, or bread. 2. Test for proteins. The Biuret test is employed to identify proteins in food samples. In this test, a solution containing copper sulfate and sodium hydroxide is added to the sample. If proteins are present, a violet color develops due to the formation of a complex between the copper ions and peptide bonds in the proteins. This color change confirms the presence of proteins in the tested food substance. 3. Test for lipids. The Sudan 3 or Sudan 4 test is used to detect the presence of lipids, particularly fats and oils, in food samples. In this test, a few drops of Sudan dye solution are added to the sample. Lipids dissolve in the Sudan dye, resulting in the formation of a distinct red-colored layer on top of the liquid. This separation confirms the presence of lipids in the food substance. 4. Test for reducing sugars. Benedict's test is employed to identify reducing sugars, such as glucose and fructose, in food samples. In this test, a food sample is mixed with Benedict's reagent, which contains copper sulfate and sodium citrate. The mixture is then heated in a water bath. If reducing sugars are present, they react with the copper ions, resulting in the formation of a color change, ranging from green to orange or brick red, depending on the concentration of reducing sugars present. Performing these tests allows us to distinguish among different food substances based on their chemical properties. These tests are valuable tools in food analysis, quality control, and nutritional assessment. By understanding the principles behind each test and interpreting the results, we can identify the presence of starch, proteins, lipids, or reducing sugars, contributing to a deeper understanding of the composition and characteristics of various food samples. Exploring enzyme activity, investigating the impact of temperature and pH on catalase or amylase enzymes, OR, millimeter, AI. In this section, we will delve into the fascinating world of enzymes by investigating the effects of temperature and pH on the activity of catalase or amylase enzymes. Enzymes play a critical role in facilitating biochemical reactions in living organisms, and understanding how external factors influence their activity provides valuable insights into enzyme functionality. By providing a detailed explanation, we can explore the experimental investigations and their significance in enzyme research. 1. Enzymes and their role in biochemical reactions. Enzymes are biological catalysts that speed up chemical reactions within living organisms. Catalase and amylase are examples of enzymes involved in specific biochemical processes. Catalase aids in the breakdown of hydrogen peroxide into water and oxygen, while amylase assists in the hydrolysis of starch into smaller sugar molecules. Investigating the factors that affect enzyme activity helps us understand the optimal conditions for enzyme functionality. 2. Investigating temperature effects. One crucial factor influencing enzyme activity is temperature. Different enzymes have distinct temperature optima at which they exhibit the highest activity. By performing experiments, varying the temperature within a suitable range, and measuring the enzyme activity, we can construct temperature activity profiles. Typically, enzyme activity increases with temperature up to a point, called the optimum temperature. Beyond this temperature, enzyme activity declines due to denaturation, which alters the enzyme's structure and reduces its functionality. 3. Examining pH effects. pH, the measure of acidity or alkalinity, also affects enzyme activity. Each enzyme has an optimal pH range in which it operates most efficiently. Deviating from this range can disrupt the enzyme's structure and impact its functionality. By conducting experiments that alter the pH of the enzyme's environment and measuring the enzyme's activity, we can determine the optimal pH range. This knowledge helps in understanding the enzyme's stability and activity in different physiological or experimental conditions. 4. Experimental methodologies. To investigate the effects of temperature and pH on enzyme activity, controlled experiments are performed. The enzyme activity is typically measured through various techniques, such as monitoring the rate of reaction or measuring the amount of product produced. Experiments involve incubating the enzyme with a substrate under different temperature or pH conditions and recording the resulting enzymatic activity. Statistical analyses can then be employed to assess the significance of the observed changes. 5. Significance of enzyme activity studies. Understanding the effects of temperature and pH on enzyme activity has broad implications in various scientific disciplines. In fields such as biochemistry, molecular biology, and medicine, this knowledge contributes to enzyme characterization, drug development, and diagnostic assays. Investigating the optimal conditions for enzyme activity allows researchers to optimize reaction conditions and improve the efficiency of enzymatic processes in industrial applications. By investigating the impact of temperature and pH on the activity of catalase or amylase enzymes, we gain valuable insights into the factors that influence enzyme functionality. These experiments contribute to our understanding of enzyme kinetics, stability, and the conditions that optimize their catalytic activity. Furthermore, this knowledge has practical applications in various scientific and industrial domains, enhancing our ability to harness the power of enzymes in diverse fields of study. Skills, equipping individuals for success and adaptability in a dynamic world. In this section, we will explore the significance of skills and their role in preparing individuals for success and adaptability in a rapidly changing world. Skills are not only crucial for professional growth but also essential for personal development and navigating various aspects of life. By providing a detailed explanation, we can delve into the importance of skills and how they contribute to overall competence and lifelong learning. 1. Acquisition and development of skills. Skills are competencies that individuals acquire and develop through learning, practice, and experience. They encompass a wide range of abilities, including both technical or domain-specific skills and transferable skills that can be applied across different contexts. Acquiring and honing skills requires dedication, persistence, and a growth mindset that embraces continuous learning and improvement. 2. Importance of transferable skills. Transferable skills, also known as soft skills or employability skills, are highly valuable in various areas of life. These skills include communication, problem-solving, critical thinking, teamwork, adaptability, creativity, and leadership. Transferable skills enable individuals to navigate complex situations, collaborate effectively, think critically, and communicate their ideas clearly. These skills are often sought after by employers as they contribute to professional success and career advancement. 3. Professional competence and career development. Skills play a critical role in professional competence and career development. Possessing the necessary technical skills in a specific field equips individuals to perform tasks efficiently and effectively. Additionally, transferable skills such as communication, leadership, and problem-solving enable individuals to excel in their roles, collaborate with colleagues, and adapt to changing work environments. Continuous skill development enhances professional growth and opens up new opportunities for advancement. 4. Personal growth and well-being. Skills are not limited to professional contexts but also contribute to personal growth and well-being. Developing skills like time management, resilience, emotional intelligence, and self-awareness improves individuals' ability to handle challenges, build positive relationships, and maintain a healthy work-life balance. These skills enhance overall well-being and contribute to a fulfilling and successful life. 5. Lifelong learning and skill adaptation. Skills are not static, they need to be continuously nurtured and adapted to keep pace with evolving demands and advancements. Lifelong learning is essential to stay relevant and thrive in a rapidly changing world. The ability to learn new skills, adapt to technological advancements, and embrace continuous improvement is crucial for personal and professional growth. Recognizing the importance of skills empowers individuals to prioritize skill development alongside acquiring knowledge. The acquisition and refinement of transferable skills enable individuals to navigate complexities, adapt to change, and excel in their chosen paths. Skills contribute to competence, adaptability, personal growth, and well-being, fostering lifelong learning and preparing individuals for success in a dynamic world. Observation and recording, documenting and analyzing observations in food substance differentiation tests, OR. In this section, we will explore the significance of observation and recording skills in the context of tests aimed at distinguishing different food substances. Students will actively engage in recording their observations during these tests and subsequently analyze the results. By providing a detailed explanation, we can understand the importance of observation, accurate documentation, and critical analysis in the differentiation of food substances. 1. The role of observation in food substance differentiation. Observation plays a crucial role in food substance differentiation tests. Students carefully observe the reactions, color changes, or other visible indicators that occur during these tests. By keenly observing and documenting these observations, students can identify and differentiate various food substances based on their unique characteristics and responses to specific tests. 2. Accurate recording of observations. Accurate recording of observations is essential in maintaining a systematic record of the testing process. Students document their observations in a clear and organized manner, noting relevant details such as the appearance, color changes, or any other observable changes that occur during the tests. Accurate documentation ensures that the recorded data is reliable and can be referred back to during analysis and interpretation. 3. Analysis of recorded observations. After completing the tests and recording observations, students engage in the analysis of their findings. They review their recorded data and look for patterns, correlations, or significant differences among the tested food substances. Through critical analysis, students can identify common characteristics or specific indicators that help distinguish one food substance from another. 4. Interpreting results and drawing conclusions. Based on their analysis of the recorded observations, students can interpret the results and draw meaningful conclusions regarding the differentiation of food substances. By comparing their findings with known characteristics or expected outcomes, students can confirm the presence or absence of specific substances such as starch, proteins, lipids, or reducing sugars in the tested samples. These conclusions contribute to a deeper understanding of the composition and properties of different food substances. The process of observation and recording during food substance differentiation tests fosters scientific inquiry, attention to detail, and critical thinking skills. Accurate documentation of observations provides a comprehensive record of the testing process, allowing for thorough analysis and interpretation of the results. Through these practices, students develop skills in data analysis, pattern recognition, and drawing evidence-based conclusions. The ability to observe, record, and analyze data is fundamental not only in the context of food substance differentiation but also in broader scientific investigations and research. By honing their observation and recording skills, students develop a scientific mindset and the ability to make informed judgments based on evidence. These skills are transferable to various fields of study and contribute to the development of well-rounded individuals equipped with critical thinking and analytical capabilities. Measurement and manipulation, precise control of temperature and pH in enzyme activity experiments, millimeter. In this section, we will explore the crucial skill of measurement and manipulation in the context of enzyme activity experiments. This skill is necessary for accurately measuring and controlling temperature and pH, two key factors that influence enzyme activity. By providing a detailed explanation, we can understand the significance of precise measurement and manipulation in ensuring reliable and meaningful experimental results. 1. Importance of accurate measurement. Accurate measurement is vital in enzyme activity experiments to ensure reliable and reproducible results. For temperature, precise measurement is required to maintain the desired experimental conditions and explore the effects of different temperatures on enzyme activity. Similarly, accurate measurement of pH is essential to investigate the impact of varying acidity or alkalinity on enzyme functionality. Reliable measurements serve as a foundation for valid conclusions and further analysis. 2. Temperature measurement and control. Precise temperature control is essential in enzyme activity experiments to explore the relationship between temperature and enzyme activity. Temperature affects the rate of enzymatic reactions, and even slight variations can significantly impact the results. Therefore, using calibrated and accurate thermometers or temperature sensors, researchers measure and manipulate the temperature to maintain the desired conditions throughout the experiment. 3. pH measurement and control. pH measurement and control are crucial for investigating the effects of acidity or alkalinity on enzyme activity. Enzymes often have an optimal pH range in which they exhibit the highest activity. By using pH meters or pH indicators, researchers measure the pH of the enzyme solution and adjust it as needed to maintain the desired pH level throughout the experiment. Precise control of pH ensures that the observed effects on enzyme activity are attributable to pH variations. 4. Experimental setup and equipment. Measurement and manipulation skills involve setting up the experiment correctly and using appropriate equipment for temperature and pH control. This includes utilizing water baths, incubators, or temperature-controlled chambers to maintain a stable and consistent temperature. pH adjustments can be made using buffer solutions or acid-base solutions to achieve the desired pH level. Skilled handling of equipment and adherence to experimental protocols ensure accurate measurement and control. 5. Data integrity and validity. Accurate measurement and manipulation techniques contribute to data integrity and the validity of experimental results. Precise control of temperature and pH allows researchers to isolate the effects of these variables on enzyme activity, eliminating confounding factors. Reliable data obtained through accurate measurement and manipulation enhance the scientific rigor of the study and support the development of robust conclusions. The skill of measurement and manipulation is crucial for conducting enzyme activity experiments with precision and accuracy. Accurate measurement of temperature and pH, coupled with careful control of these variables, ensures the reliability and validity of experimental data. Through meticulous measurement and manipulation, researchers can elucidate the relationship between temperature, pH, and enzyme activity, contributing to a deeper understanding of enzymatic processes and their regulation. Developing proficiency in measurement and manipulation skills is not only applicable to enzyme activity experiments but also essential in various scientific disciplines. These skills enable researchers to conduct experiments, collect reliable data, and advance knowledge in diverse areas of study. Mastery of measurement and manipulation techniques enhances experimental precision, promotes scientific inquiry, and supports evidence-based conclusions. Analysis and interpretation, unveiling insights from experimental data and exploring the effects of temperature and pH on enzyme activity, AI. In this section, we will explore the crucial skill of analysis and interpretation in the context of enzyme activity experiments. Students play a vital role in analyzing the data collected during the experiments and interpreting the effects of temperature and pH on enzyme activity. By providing a detailed explanation, we can understand the significance of thorough analysis and interpretation in uncovering insights and drawing meaningful conclusions from experimental findings. 1. Data analysis in enzyme activity experiments. Data analysis involves examining and evaluating the collected data to uncover patterns, trends, and relationships. In enzyme activity experiments, students analyze the data, which may include measurements of enzyme activity, reaction rates, or other relevant parameters recorded under varying temperature and pH conditions. Through statistical analysis or graphical representations, students identify key observations and patterns within the dataset. 2. Exploring the effects of temperature on enzyme activity. Analysis and interpretation enable students to understand the impact of temperature on enzyme activity. By examining the experimental data collected at different temperatures, students can identify trends in enzyme activity. For example, they may observe an increase in enzyme activity up to an optimal temperature, beyond which the activity decreases due to enzyme denaturation. Through analysis, students can determine the temperature range in which the enzyme exhibits the highest activity. 3. Investigating the effects of pH on enzyme activity. The skill of analysis and interpretation helps students explore the influence of pH on enzyme activity. By analyzing the experimental data collected at different pH levels, students can observe how changes in acidity or alkalinity affect enzyme functionality. They may discover an optimal pH range in which the enzyme demonstrates the highest activity. Deviations from this range may result in reduced enzyme activity due to alterations in the enzyme's structure or active site. 4. Drawing meaningful conclusions. Through comprehensive analysis and interpretation, students draw meaningful conclusions regarding the effects of temperature and pH on enzyme activity. By considering the trends and patterns observed in the data, they can infer how changes in these variables impact the enzymatic process. Additionally, students can discuss the mechanisms underlying these effects, such as enzyme-substrate interactions, protein stability, or protonation state changes, depending on the specific enzyme being studied. 5. Implications and further exploration. The skill of analysis and interpretation extends beyond the immediate experiment, providing insights and opening avenues for further exploration. Students can discuss the practical implications of their findings, such as the relevance of temperature and pH optimization in industrial processes or the potential impact on enzyme functionality in different biological systems. The ability to critically analyze and interpret data lays the foundation for future research and understanding of enzymatic mechanisms. Analysis and interpretation skills are integral to the scientific process, enabling students to make sense of experimental findings and draw evidence-based conclusions. By analyzing data, students uncover insights into the effects of temperature and pH on enzyme activity, deepening their understanding of enzymatic processes and the factors that influence them. Through interpretation, students bridge the gap between raw data and meaningful conclusions, fostering critical thinking and scientific inquiry. Proficiency in analysis and interpretation skills empowers students to explore the intricacies of enzymology and apply their knowledge in various scientific disciplines. These skills enhance their ability to analyze experimental data, contribute to the scientific community's understanding, and drive advancements in enzymatic research. Respiration, unveiling the intricacies of cellular energy production. Respiration, an essential biological process, involves the conversion of organic molecules into usable energy by living organisms. In this section, we will delve into the multifaceted nature of respiration, exploring its different stages, mechanisms, and significance in cellular energy production. By providing a detailed explanation, we can deepen our understanding of this vital process and its intricate workings within living organisms. 1. Overview of respiration. Respiration is a series of metabolic reactions that occur within cells to extract energy from organic molecules, typically carbohydrates. It encompasses three main stages: glycolysis, the Krebs cycle, also known as the citric acid cycle or TCA cycle, and oxidative phosphorylation, including the electron transport chain. Together, these stages result in the production of adenosine triphosphate, ATP, the primary energy currency of cells. 2. Glycolysis, breaking down glucose. Glycolysis, the initial stage of respiration, occurs in the cytoplasm of cells. It involves the breakdown of glucose, a 6-carbon molecule, into 2 molecules of pyruvate, a 3-carbon compound. Throughout this process, a small amount of ATP is generated, and high-energy electrons and hydrogen ions are transferred to carrier molecules, such as in ADH. 3. The Krebs cycle, extracting energy from pyruvate. The Krebs cycle takes place in the mitochondria of eukaryotic cells. In this stage, each pyruvate molecule undergoes a series of reactions, releasing carbon dioxide and transferring high-energy electrons to carrier molecules, NADH and FADH2. The Krebs cycle generates additional ATP and forms reduced coenzymes, which carry high-energy electrons to the next stage. 4. Oxidative phosphorylation, electron transport chain and ATP synthesis. Oxidative phosphorylation occurs in the inner mitochondrial membrane and involves the electron transport chain, etc., and ATP synthesis. The reduced coenzymes, NADH and FADH2, from previous stages donate their high-energy electrons to the etc., which generates a proton gradient across the membrane. This proton gradient drives ATP synthesis through the action of ATP synthase, resulting in the production of a significant amount of ATP. 5. Significance of respiration. Respiration is vital for sustaining life as it provides the necessary energy for cellular processes, growth, and maintenance. Field or profession and are acquired through formal education, training programs, or hands-on experience. These skills are essential for performing specialized tasks and functions within a specific domain. On the other hand, transferable skills, also known as soft skills or employability skills, are highly valuable across different areas of life. They include communication, teamwork, problem-solving, critical thinking, adaptability, leadership, and many others. Transferable skills enable individuals to effectively navigate challenges, work collaboratively, think critically, and communicate their ideas clearly. These skills are sought after by employers as they contribute to professional success and career advancement. Skills play a pivotal role in personal growth and development. By acquiring and honing various skills, individuals enhance their abilities to manage time effectively, communicate with clarity, adapt to changing circumstances, and handle conflicts. Skills such as resilience, emotional intelligence, and self-awareness contribute to mental well-being and foster healthy relationships. Additionally, skills in creativity, innovation, and entrepreneurial thinking promote personal fulfillment and facilitate the pursuit of passions and interests. In the professional sphere, skills are instrumental in career development and success. Possessing the necessary technical skills within a specific field equips individuals to perform tasks efficiently and effectively. Additionally, transferable skills such as leadership, problem-solving, and adaptability enable individuals to excel in their roles, collaborate with colleagues, and navigate dynamic work environments. Continuous skill development and staying abreast of emerging trends enhance professional growth and open up new opportunities for advancement. It is important to recognize that skills are not static, they require continuous nurturing and adaptation to keep pace with evolving demands and advancements. Lifelong learning is essential to stay relevant and thrive in a rapidly changing world. The ability to learn new skills, embrace technological advancements, and adapt to shifting professional landscapes is crucial for maintaining professional competitiveness and personal growth. Investing in skill development and acquisition is a lifelong endeavor that empowers individuals to unlock their full potential. By honing their skills, individuals develop confidence, competence, and a growth mindset. Skills enable individuals to tackle challenges, seize opportunities, and contribute meaningfully to their communities and organizations. Furthermore, skills foster resilience, adaptability, and a sense of empowerment, equipping individuals with the tools needed to navigate the complexities of an ever-changing world. By recognizing the significance of skills and actively developing them, individuals enhance their personal and professional capabilities, opening doors to new possibilities and creating pathways for success. Skills are not merely a means to an end but an essential component of personal and professional growth, facilitating lifelong learning, adaptability, and the pursuit of excellence in various domains of life. Observation and recording, documenting and analyzing the products of anaerobic respiration in yeast and the effects of external factors on transpiration, OR. In this section, we will explore the significance of observation and recording skills in the context of studying anaerobic respiration in yeast and the effects of external factors on transpiration. Students play an active role in observing and documenting the products of anaerobic respiration in yeast and the changes in transpiration rates influenced by external factors. By providing a detailed explanation, we can understand the importance of careful observation, accurate documentation, and data analysis in uncovering valuable insights. 1. Observing the products of anaerobic respiration in yeast. Observation skills are critical for studying the products of anaerobic respiration in yeast. During controlled experiments, students closely observe the fermentation process in yeast, noting any visible changes or indicators of metabolic activity. These observations may include the release of gas bubbles, changes in color or texture, or the formation of byproducts such as ethanol. Accurate and detailed observations provide valuable information about the metabolic pathways involved in anaerobic respiration. 2. Recording and documenting observations. Accurate recording and documentation are essential in capturing the observations made during the experiments. Students carefully record their findings, noting the specific conditions of the experiments, such as the type and concentration of substrates used, the duration of the experiment, and any relevant environmental factors. Detailed documentation ensures that the recorded data is reliable and can be referred back to during analysis and interpretation. 3. Analyzing the effects of external factors on transpiration. In the context of studying transpiration, observation and recording skills are also crucial for analyzing the effects of external factors. Students observe changes in transpiration rates under different conditions of light intensity, temperature, humidity, and air movements. By accurately documenting these changes, students can analyze the data and identify patterns or trends related to the influence of external factors on transpiration. 4. Data analysis and interpretation. After completing the observations and recording data, students engage in the analysis and interpretation of their findings. They review their recorded data, compare and contrast the observations made under different experimental conditions, and identify correlations between external factors and transpiration rates. Data analysis techniques, such as graphical representations or statistical tests, may be employed to identify significant differences and draw meaningful conclusions. 5. Drawing conclusions and validating findings. Based on the analysis of recorded observations and data, students draw conclusions about the products of anaerobic respiration in yeast and the effects of external factors on transpiration. These conclusions are supported by evidence obtained through careful observation, accurate recording, and thorough data analysis. Validating findings through consistent patterns and statistical significance enhances the reliability and credibility of the conclusions drawn. The skill of observation and recording, coupled with data analysis and interpretation, is integral to scientific inquiry. By documenting observations and analyzing data, students contribute to the body of knowledge in anaerobic respiration and transpiration. These skills foster critical thinking, attention to detail, and the ability to make evidence-based conclusions. The ability to accurately observe, record, and analyze data is not limited to the laboratory setting but extends to various scientific disciplines and real-world applications. Developing proficiency in these skills equips students with a scientific mindset and prepares them for future scientific investigations, research endeavors, and evidence-based decision-making. Observation and recording skills are foundational in the scientific process, providing a systematic approach to gathering and analyzing data. By honing these skills, students develop a deeper understanding of the phenomena under investigation, contribute to scientific knowledge, and cultivate the essential skills needed for scientific exploration and discovery. Measurement and manipulation, precise control of factors in investigative studies, millimeter. In this section, we will explore the significance of measurement and manipulation skills in the context of investigative studies. Students play a crucial role in measuring and controlling factors such as light intensity, temperature, humidity, and air movements during their investigations. By providing a detailed explanation, we can understand the importance of precise measurement and manipulation in ensuring accurate and reliable experimental results. 1. Importance of accurate measurement. Accurate measurement is vital in investigative studies to ensure reliable and meaningful results. Students carefully measure factors such as light intensity, temperature, humidity, and air movements using appropriate instruments and techniques. Precise measurements provide a foundation for understanding the relationships between these factors and the phenomena under investigation. Accurate measurement allows for comparisons, analysis, and drawing reliable conclusions. 2. Control of factors. In investigative studies, students exercise control over factors such as light intensity, temperature, humidity, and air movements. By manipulating these factors, students can investigate their influence on specific phenomena or processes. Controlling factors allows for the isolation of variables, ensuring that any observed effects can be attributed to the manipulated factor under investigation. Precise control enhances the reliability and validity of experimental results. 3. Measurement tools and techniques. Measurement and manipulation skills involve the use of appropriate tools and techniques. Students employ instruments such as light meters, thermometers, hygrometers, anemometers, or specialized sensors to measure and quantify factors such as light intensity, temperature, humidity, and air movements. By selecting and using these tools correctly, students obtain accurate measurements and facilitate the controlled manipulation of these factors. 4. Experimental setup and protocols. Measurement and manipulation skills encompass setting up experiments and adhering to established protocols. Students design controlled environments or experimental setups where factors can be precisely measured and manipulated. They follow standardized procedures to ensure consistency across experiments, minimizing sources of error and facilitating accurate data collection. 5. Data integrity and validity. Accurate measurement and manipulation techniques contribute to data integrity and the validity of experimental results. Precise control of factors allows for the accurate quantification and manipulation of variables, reducing confounding influences and increasing the reliability of the data. By employing robust measurement and manipulation practices, students enhance the scientific rigor of their investigations, fostering confidence in the results obtained. Measurement and manipulation skills are fundamental not only in the context of specific investigations but also in broader scientific endeavors. The ability to accurately measure and control factors is essential in conducting experiments, collecting reliable data, and drawing meaningful conclusions. These skills foster scientific inquiry, attention to detail, and critical thinking, providing a solid foundation for further research and scientific exploration. Developing proficiency in measurement and manipulation skills equips students with the ability to design and conduct controlled experiments, investigate cause and effect relationships, and contribute to scientific knowledge. These skills extend beyond the classroom and have practical applications in various scientific fields and real-world contexts. Mastery of measurement and manipulation techniques enhances experimental precision, promotes scientific inquiry, and supports evidence-based decision-making. Analysis and interpretation, unveiling insights through data analysis and results interpretation, AI. In this section, we will explore the significance of analysis and interpretation skills in the context of investigations. Students play a vital role in analyzing the data they collect and interpreting the results of their investigations. By providing a detailed explanation, we can understand the importance of thorough data analysis and results interpretation in uncovering valuable insights and drawing meaningful conclusions. 1. Data analysis. Data analysis involves the examination and evaluation of the collected data to reveal patterns, trends, and relationships. Students employ various techniques, such as statistical analysis, graphical representations, or qualitative assessments, to analyze the data they have gathered during their investigations. Through data analysis, students identify key observations, patterns, and trends within the dataset. 2. Interpretation of results. Results interpretation involves making sense of the analyzed data and drawing meaningful conclusions. Students carefully interpret the results of their investigations by considering the patterns and trends identified during data analysis. This process involves comparing the results with established theories or expected outcomes, examining potential limitations or sources of error, and drawing evidence-based conclusions from the analyzed data. 3. Drawing inferences and insights. Through data analysis and results interpretation, students draw inferences and gain valuable insights into the phenomenon or process under investigation. They identify relationships between variables, explore cause and effect relationships, or make predictions based on the observed patterns. Results interpretation enables students to move beyond the raw data and uncover meaningful information that contributes to their understanding of the investigated topic. 4. Identifying trends and patterns. During analysis and interpretation, students identify trends and patterns in the data that emerge from their investigations. These trends may include relationships between variables, changes over time, or correlations between different factors. By recognizing these patterns, students gain a deeper understanding of the phenomenon under study and can formulate hypotheses or further questions for future investigations. 5. Validation and evaluation. Analysis and interpretation involve the validation and evaluation of the results obtained. Students critically assess the reliability and validity of their findings, considering factors such as the quality of data collection, potential sources of bias or error, and the consistency of results with existing knowledge or theories. Through rigorous evaluation, students ensure the credibility and integrity of their results and conclusions. 6. Communication and presentation. Effective analysis and interpretation skills extend to the ability to communicate and present the results of investigations. Students articulate their findings, interpretations, and insights in a clear and organized manner. They employ appropriate scientific language, visual aids, and supporting evidence to convey their conclusions effectively. Strong communication skills allow students to share their discoveries, engage in scientific discussions, and contribute to the broader scientific community. Analysis and interpretation skills are fundamental to the scientific process, enabling students to make sense of their data and draw evidence-based conclusions. Through data analysis, results interpretation, and drawing inferences, students unlock insights, deepen their understanding, and contribute to scientific knowledge in their respective fields of investigation. Developing proficiency in analysis and interpretation skills empowers students to critically analyze and interpret data, assess the significance of their findings, and communicate their conclusions effectively. These skills extend beyond the specific investigations and have applications in various scientific disciplines and real-world contexts. Mastery of analysis and interpretation techniques enhances the scientific rigor of investigations, fosters critical thinking, and supports evidence-based decision-making. Planning and designing, strategizing and structuring controlled investigations on respiration and transpiration, PD. In this section, we will explore the importance of planning and designing skills in the context of conducting controlled investigations related to respiration and transpiration. The skill of planning and designing is crucial for strategizing and structuring experiments that effectively explore these physiological processes. By providing a detailed explanation, we can understand the significance of meticulous planning, thoughtful design, and methodical implementation in conducting meaningful investigations. 1. Importance of meticulous planning. Meticulous planning is essential in conducting controlled investigations. Students engage in careful thought and consideration to develop a comprehensive plan that outlines the objectives, procedures, and resources required for the investigation. Planning ensures that all necessary components are accounted for, including the identification of research questions, determination of variables, establishment of controls, and selection of appropriate measurement techniques. Thorough planning sets the stage for a well-structured and organized investigation. 2. Designing controlled experiments. Designing controlled experiments is a key aspect of planning and designing skills. Students devise experimental designs that allow for the manipulation of variables and the control of potential confounding factors. The design ensures that any observed effects or changes can be attributed to the specific factors being investigated, such as the impact of external conditions on respiration or transpiration. Thoughtful experimental design enhances the reliability and validity of the investigation's outcomes. 3. Defining research questions and hypotheses. Planning and designing involve defining clear research questions and formulating hypotheses to guide the investigation. Students articulate specific inquiries they seek to address and develop hypotheses that predict expected outcomes based on existing knowledge or observations. Well-defined research questions and hypotheses provide a focused direction for the investigation and facilitate the collection of relevant data. 4. Consideration of variables and controls. Planning and designing skills encompass identifying and considering variables and controls in the investigation. Students determine the key factors that may influence the processes of respiration and transpiration and develop strategies to manipulate and control them systematically. Variables that are not of immediate interest or influence on the investigation are controlled to ensure that observed changes or effects can be attributed to the factors being investigated. 5. Selection of measurement techniques and tools. Planning and designing also involves selecting appropriate measurement techniques and tools for data collection. Students identify and choose methods and instruments that enable accurate and precise measurements of relevant variables. The selection of measurement techniques is guided by the nature of the investigation, the characteristics of the phenomena being studied, and the availability of suitable equipment. 6. Ethical considerations and safety measures. Planning and designing include addressing ethical considerations and implementing safety measures. Students take into account ethical guidelines and considerations, ensuring the responsible and respectful treatment of organisms and resources involved in the investigation. They also incorporate safety protocols and procedures to safeguard themselves and others involved in the experiment, ensuring a secure and controlled environment. Effective planning and designing skills are vital for the successful execution of controlled investigations related to respiration and transpiration. Meticulous planning ensures that investigations are purposeful, well-structured, and scientifically rigorous. Thoughtful experimental design allows for the systematic exploration of variables and the control of potential confounders, enabling students to draw meaningful conclusions and make evidence-based claims. Proficiency in planning and designing skills equips students with the ability to strategize investigations, formulate research questions, develop hypotheses, and implement systematic methodologies. These skills extend beyond the immediate investigations and have practical applications in various scientific disciplines and real-world contexts. Mastery of planning and designing techniques enhances scientific inquiry, fosters critical thinking, and supports the formulation and execution of well-controlled experiments. Transport, the dynamic movement of substances in biological systems. Transport refers to the process of moving substances within biological systems, enabling the efficient distribution of essential molecules, nutrients, gases, and waste products. In this section, we will explore the intricate mechanisms and significance of transport in various biological contexts. By providing a detailed explanation, we can delve into the different types of transport, their underlying processes, and their crucial roles in maintaining cellular functions and overall organismal homeostasis. 1. Overview of transport. Transport is a fundamental biological process that occurs at various scales, from the movement of molecules within individual cells to the circulation of fluids in multicellular organisms. It encompasses a diverse range of mechanisms and systems that facilitate the transport of substances across cellular membranes, through circulatory systems, or within specialized tissues and organs. The efficient transport of molecules is essential for supporting cellular metabolism, growth, and overall organismal function. 2. Cellular transport. At the cellular level, transport processes occur across cell membranes, enabling the exchange of molecules between the internal and external environments. Various mechanisms govern cellular transport, including passive diffusion, facilitated diffusion, active transport, endocytosis, and exocytosis. These mechanisms ensure the controlled movement of ions, nutrients, signaling molecules, and waste products, maintaining cellular homeostasis and enabling proper cell function. 3. Circulatory transport. In multicellular organisms, circulatory systems play a vital role in transporting substances throughout the body. The cardiovascular system, composed of the heart, blood vessels, and blood, facilitates the circulation of oxygen, nutrients, hormones, and metabolic waste products. Blood, propelled by the pumping action of the heart, transports these substances, allowing them to reach the cells and tissues that require them for proper functioning. 4. Specialized transport systems. In addition to cellular and circulatory transport, specialized transport systems exist to facilitate the movement of specific substances within organisms. Examples include the respiratory system, responsible for the exchange of gases such as oxygen and carbon dioxide, the digestive system, involved in the absorption of nutrients from food, and the renal system, responsible for the filtration and reabsorption of waste products and the regulation of water and ion balance. These specialized systems ensure the efficient transport of substances to meet the specific needs of different tissues and organs. 5. Transport across membranes. The movement of substances across cellular membranes is a critical aspect of transport. It occurs through various mechanisms, depending on the properties of the molecules involved and the concentration gradients across the membrane. Passive processes such as diffusion and osmosis allow substances to move along their concentration gradients, while active transport requires energy expenditure to move molecules against their concentration gradients. Transport proteins, such as channels, carriers, and pumps, play essential roles in facilitating these processes. 6. Regulation of transport. Transport processes are tightly regulated to maintain homeostasis and meet the dynamic needs of the organism. Regulatory mechanisms ensure that the transport of substances is balanced and responsive to changing physiological conditions. Feedback loops, hormonal control, and cellular signaling pathways help modulate transport rates and adjust them according to metabolic demands, environmental cues, and internal states. Understanding the intricacies of transport processes is essential in fields such as physiology, cellular biology, and biomedical sciences. It sheds light on how organisms efficiently exchange molecules and maintain internal balance. By studying transport, researchers can explore the mechanisms underlying nutrient uptake, gas exchange, waste elimination, and the interplay between transport systems and other physiological processes. Transport serves as a cornerstone of biological systems, enabling the delivery of essential molecules, removal of waste products, and maintenance of homeostasis. Its comprehension deepens our understanding of the intricate processes that sustain life and provides a foundation for further exploration of cellular and organismal functions. Drawing and labeling diagrams, illustrating the structures of arteries, veins, and capillaries, Dr. In this section, we will explore the significance of drawing and labeling diagrams as a means to visually represent the structures of arteries, veins, and capillaries. By providing a detailed explanation, we can understand the importance of accurate and well-labeled diagrams in enhancing our understanding of the anatomical features and functions of these blood vessels. 1. Visual representation through diagrams. Diagrams provide a visual representation of anatomical structures, enabling a clear and concise depiction of the features and relationships within complex systems such as the circulatory system. By drawing diagrams of arteries, veins, and capillaries, we can visualize the unique characteristics of these blood vessels, including their sizes, shapes, branching patterns, and connections. 2. Anatomical structures of arteries. Arteries are blood vessels that carry oxygenated blood away from the heart and distribute it to various tissues and organs throughout the body. When drawing and labeling diagrams of arteries, it is important to highlight their distinguishing features. These include thick and elastic walls that allow for the accommodation of high blood pressure and the presence of smooth muscle fibers that aid in the regulation of blood flow. Arteries typically exhibit a more circular or tubular shape compared to other blood vessels. 3. Anatomical structures of veins. Veins are blood vessels that transport deoxygenated blood back to the heart from the tissues and organs. Diagrams of veins should depict their unique characteristics. Veins have thinner walls compared to arteries and are equipped with valves that prevent backflow of blood and facilitate the unidirectional flow towards the heart. Veins often appear flatter and less rigid in their structure, allowing them to be easily compressed and expanded. 4. Anatomical structures of capillaries. Capillaries are the smallest blood vessels and serve as the site of exchange between the blood and surrounding tissues. Diagrams illustrating capillaries should highlight their microscopic and delicate nature. Capillaries consist of a single layer of endothelial cells, allowing for the diffusion of oxygen, nutrients, and waste products between the blood and tissues. The extensive network of capillaries ensures that every cell within the body is in close proximity to these vital exchange points. 5. Importance of labeling. Labeling the structures within the diagrams of arteries, veins, and capillaries is crucial for enhancing comprehension. Clear and accurate labeling of key components, such as the endothelium, smooth muscle layers, valves, in veins, and connections to the heart, helps to identify and understand the specific anatomical features and functions of each blood vessel type. Proper labeling enables viewers to gain a comprehensive understanding of the structures being depicted. Drawing and labeling diagrams of arteries, veins, and capillaries provide a visual representation that aids in the understanding of their anatomical features and functions. These diagrams help learners, students, and researchers comprehend the unique characteristics of each blood vessel type and their role in the circulatory system. Accurate representation and labeling of these structures promote a solid foundation for further exploration of cardiovascular physiology, pathology, and medical education. By visually depicting the intricate structures of arteries, veins, and capillaries, diagrams facilitate the comprehension of their distinct properties and functions within the circulatory system. These visual representations serve as valuable learning tools, enhancing our understanding of the complex network of blood vessels and their essential role in maintaining proper blood circulation throughout the body. Examining mammalian hearts, exploring external and internal features, OR. In this section, we will delve into the significance of examining mammalian hearts and the valuable insights gained by exploring their external and internal features. By providing a detailed explanation, we can understand the importance of careful observation and analysis in uncovering the anatomical structures and physiological functions of these vital organs. 1. External examination. External examination involves observing and analyzing the visible features of mammalian hearts. This includes examining the size, shape, and overall structure of the heart. By carefully examining the external features, such as the chambers, major blood vessels, and surfaces, we can gain initial insights into the complexity and organization of the heart. 2. Observation of chambers and valves. The heart is comprised of 4 chambers, 2 atria and 2 ventricles. By examining the external features, we can identify these chambers and their relative positions. Furthermore, the examination of valves, such as the atrioventricular, AV, valves and semilunar valves, helps to understand their roles in regulating the flow of blood between chambers and out of the heart. 3. Analysis of coronary vessels. Mammalian hearts are supplied with oxygenated blood through coronary vessels. During external examination, it is valuable to identify these vessels and their pathways on the surface of the heart. The coronary arteries deliver oxygenated blood to the heart muscle, while the coronary veins collect deoxygenated blood and return it to the circulation. Analyzing these vessels provides insights into the vital blood supply that nourishes the cardiac tissue. 4. Internal examination. Internal examination involves dissecting the mammalian heart to explore its internal structures. This provides a more detailed understanding of its complex anatomy. By carefully dissecting and observing the internal features, such as the chambers, valves, septa, and papillary muscles, we can gain a comprehensive view of the intricate organization of the heart. 5. Identification of cardiac muscle layers. The walls of the heart are composed of specialized cardiac muscle tissue. During internal examination, it is essential to identify and analyze the layers of cardiac muscle. The myocardium, the thickest layer, is responsible for the forceful contraction of the heart, while the endocardium and epicardium contribute to the smooth inner lining and protective outer layer of the heart, respectively. 6. Exploration of internal structures. By dissecting the internal structures, we can explore the intricate arrangement of valves, chordae tendineae, and papillary muscles within the chambers. This examination allows us to understand how these structures function together to ensure proper blood flow, prevent backflow, and maintain the rhythmic pumping action of the heart. Examining external and internal features of mammalian hearts through careful observation and analysis provides valuable insights into their anatomical structures and physiological functions. This examination allows us to appreciate the complexity and organization of the heart, including its chambers, valves, coronary vessels, and specialized cardiac muscle layers. Understanding the intricate details of the heart enhances our comprehension of cardiovascular physiology, cardiac pathology, and the interplay between structure and function in these remarkable organs. By examining mammalian hearts, researchers, students, and medical professionals gain a deeper understanding of the unique features and complexities of these vital organs. This examination serves as a foundation for further exploration of cardiac anatomy, function, and the diagnosis and treatment of cardiac conditions. The knowledge gained from examining mammalian hearts contributes to advancements in cardiovascular research, patient care, and the overall understanding of the intricacies of the circulatory system. Skills, empowering individuals with abilities to thrive and succeed. Skills are a set of acquired abilities that enable individuals to perform tasks, solve problems, and achieve goals effectively. In this section, we will explore the significance of skills and their impact on personal and professional development. By providing a detailed explanation, we can understand how skills contribute to individual growth, enhance performance, and open doors to opportunities. 1. The value of skills. Skills play a pivotal role in personal and professional success. They encompass a wide range of competences, including technical skills specific to a particular field or profession and transferable skills that can be applied across different contexts. Skills are the building blocks that enable individuals to navigate challenges, achieve desired outcomes, and adapt to changing circumstances. They provide individuals with the tools needed to excel in various areas of life. 2. Technical skills. Technical skills are specialized abilities acquired through formal education, training, or hands-on experience in a specific field. These skills are often specific to particular industries or professions and are essential for performing specific tasks or functions. Examples of technical skills include programming, medical expertise, engineering proficiency, and artistic craftsmanship. Technical skills are vital for professional growth and career advancement. 3. Transferable skills. Transferable skills, also known as soft skills or general competences, are versatile abilities that can be applied across different roles, industries, and situations. These skills include communication, teamwork, problem-solving, critical thinking, adaptability, leadership, and emotional intelligence, among others. Transferable skills are highly sought after by employers as they contribute to effective collaboration, innovation, and success in diverse work environments. 4. Continuous skill development. Skills are not static, they require continuous development and refinement. Lifelong learning is essential for staying relevant, adapting to new technologies and advancements, and seizing new opportunities. Individuals who actively invest in their skill development through continuous learning, training programs, and professional development enhance their capabilities and remain competitive in their respective fields. 5. Skill application in various contexts. Skills are not limited to specific domains but can be applied across various contexts. For example, effective communication skills are valuable in personal relationships, team collaborations, customer service, and leadership roles. Problem-solving skills are crucial in overcoming challenges both at work and in personal life. The ability to adapt and embrace change is essential for navigating uncertainties and seizing opportunities in any context. 6. Skill enhancement and mastery. Developing skills requires deliberate practice, feedback, and a growth mindset. By actively seeking opportunities to apply and enhance their skills, individuals can progress from novice to mastery level. Continued practice, seeking constructive feedback, and embracing a growth mindset enable individuals to refine their abilities and continuously improve their performance. Skills empower individuals to pursue their passions, achieve personal goals, and thrive in the professional realm. They foster personal growth, boost self-confidence, and contribute to overall well-being. Additionally, possessing a diverse range of skills expands opportunities, as individuals can adapt to different roles, industries, and ever-evolving job market demands. Recognizing the importance of skills and actively developing them opens doors to personal and professional growth. It enables individuals to unlock their full potential, seize new opportunities, and make meaningful contributions to their communities and organizations. By nurturing and expanding their skill set, individuals equip themselves with the capabilities needed to overcome challenges, achieve success, and lead fulfilling lives. Drawing, visualizing and understanding the anatomical features of arteries, veins, capillaries, and the heart, Dr. In this section, we will explore the significance of drawing as a tool for visualizing and understanding the anatomical features of arteries, veins, capillaries, and the heart. By providing a detailed explanation, we can understand how drawing enables students to grasp the intricate structures and relationships within these vital components of the circulatory system. 1. Drawing as a visual learning tool. Drawing serves as a powerful visual learning tool that allows students to create representations of anatomical structures. Through drawing, students engage in a hands-on process of observation, analysis, and representation, facilitating a deeper understanding of the complex structures of arteries, veins, capillaries, and the heart. By visually depicting these structures, students can better comprehend their spatial relationships and functional significance. 2. Visualizing arterial structures. Drawing enables students to visually represent and understand the anatomical features of arteries. They can depict the size, branching patterns, and connections of arteries, emphasizing their distinct characteristics. By drawing and labeling arterial structures, students gain insights into how these blood vessels transport oxygenated blood away from the heart to various tissues and organs, ensuring efficient delivery of nutrients and removal of waste products. 3. Depicting venous structures. Drawing allows students to portray the anatomical features of veins. They can illustrate the size, course, and connections of veins, highlighting their unique characteristics. Through drawing and labeling, students comprehend how veins transport deoxygenated blood back to the heart from tissues and organs, aiding in the removal of metabolic waste products and facilitating the return of blood for oxygenation. 4. Illustrating capillary networks. Drawing facilitates the depiction of capillary networks, the intricate web-like structures where gas and nutrient exchange occurs between blood and tissues. Students can represent the tiny, thin-walled capillaries that connect arteries and veins, emphasizing their vast network and proximity to individual cells. Through drawing, students gain a visual understanding of how capillaries provide an extensive surface area for efficient exchange of oxygen, nutrients, and waste products. 5. Visual representation of the heart. Drawing enables students to visualize and represent the anatomical structures of the heart. They can illustrate the chambers, valves, major blood vessels, and external features of the heart. By drawing and labeling the heart structures, students develop a comprehensive understanding of its complex organization, including the role of each chamber, the function of valves in regulating blood flow, and the connections to arteries and veins. Drawing and labeling structures of arteries, veins, capillaries, and the heart provide students with a visual representation that enhances their understanding of the anatomical features and functional significance of these critical components of the circulatory system. Through accurate and well-labeled drawings, students develop a deeper appreciation for the complexities of the circulatory system and how these structures work together to ensure the proper functioning of the cardiovascular system. By engaging in the process of drawing and labeling, students strengthen their observational skills, improve their spatial reasoning, and enhance their knowledge retention. Additionally, drawing fosters artistic expression and creativity while promoting a multidimensional understanding of anatomical structures. The ability to visually represent and comprehend these intricate structures equips students with a solid foundation for further exploration of cardiovascular physiology, anatomical studies, and medical education. Drawing not only aids in the understanding of anatomical features but also serves as a valuable tool for communication and collaboration among healthcare professionals, researchers, and educators. Accurate drawings and labels facilitate clear and effective communication of anatomical concepts and findings, promoting accurate diagnoses, research advancements, and educational materials. Observation and recording. Exploring and documenting the external and internal features of mammalian hearts, OR. In this section, we will delve into the significance of observation and recording as essential skills for exploring and documenting the external and internal features of mammalian hearts. By providing a detailed explanation, we can understand the importance of keen observation, precise documentation, and thorough analysis in gaining insights into the anatomical structures and physiological characteristics of these vital organs. 1. Keen observation. Keen observation is a fundamental aspect of exploring mammalian hearts. Students carefully observe the external and internal features of the heart, paying attention to details such as size, shape, color, texture, and overall appearance. By honing their observation skills, students can identify distinct structures, patterns, and variations within different hearts, enabling a comprehensive understanding of their unique characteristics. 2. External feature observation. Observing the external features of mammalian hearts involves examining the heart surface, dimensions, and anatomical landmarks. Students carefully observe and document characteristics such as the presence of blood vessels, coronary arteries and veins, fat deposits, and fibrous tissues. This observation provides insights into the overall shape and topography of the heart, enhancing the understanding of its external anatomy. 3. Internal feature observation. Internal feature observation entails the exploration and documentation of the intricate structures within the heart. Students dissect the heart to observe the chambers, valves, septa, papillary muscles, and other internal components. By closely examining these structures, students gain a deeper understanding of how the heart functions, how blood flows through the chambers, and how valves ensure unidirectional blood flow. Precise recording of these observations is crucial for accurate documentation and future reference. 4. Documenting findings. Recording observations is a critical aspect of the observation and recording process. Students document their findings, both qualitative and quantitative, using written descriptions, sketches, diagrams, or digital imaging. Precise documentation allows for the creation of comprehensive records that can be reviewed, analyzed, and shared with others. Accurate and detailed records serve as valuable references for further analysis, research, and educational purposes. 5. Analysis and interpretation. Analysis and interpretation of the recorded observations provide deeper insights into the anatomical structures and physiological characteristics of mammalian hearts. Students analyze the relationships between different structures, observe variations between specimens, and interpret their significance in the context of cardiac function. Through careful analysis, students can uncover patterns, make comparisons, and draw evidence-based conclusions about the observed features. Observation and recording skills are fundamental for gaining a comprehensive understanding of mammalian hearts. By honing these skills, students develop the ability to capture precise details, document their observations systematically, and engage in critical analysis. This process enables them to uncover the intricate complexities of the heart, its anatomical variations, and potential correlations with cardiac function and health. The observation and recording of external and internal features of mammalian hearts contribute to the advancement of knowledge in the fields of anatomy, physiology, cardiology, and medical research. Accurate documentation and detailed records facilitate the sharing of information, collaboration among researchers and healthcare professionals, and the development of improved diagnostic and treatment approaches for cardiovascular conditions. By cultivating observation and recording skills, students develop a scientific mindset, attention to detail, and the ability to critically analyze and interpret their findings. These skills extend beyond the study of mammalian hearts and have broader applications in scientific investigations, research methodologies, and evidence-based decision-making in various scientific disciplines. Excretion, removing waste products for homeostasis and health. Excretion is a vital process in living organisms that involves the removal of waste products and toxic substances to maintain internal balance and promote overall health. In this section, we will explore the significance of excretion and its role in maintaining homeostasis. By providing a detailed explanation, we can understand the mechanisms and importance of excretion in various organisms. 1. Overview of excretion. Excretion is the biological process by which waste products, metabolic byproducts, and other harmful substances are eliminated from the body. It ensures the removal of excess water, salts, nitrogenous wastes, such as urea and ammonia, and other toxic compounds. Through excretion, organisms maintain internal balance, prevent the accumulation of harmful substances, and support the proper functioning of vital organs. 2. Excretory organs and structures. Excretion involves specialized organs and structures responsible for waste removal. In humans, the kidneys are the primary excretory organs, playing a crucial role in filtering blood, regulating fluid balance, and excreting waste products in the form of urine. Other excretory structures include the lungs, which eliminate carbon dioxide and water vapor during respiration, and the skin, which excretes sweat containing metabolic waste products and helps regulate body temperature. 3. Kidney function and urine formation. The kidneys play a central role in excretion by filtering waste materials from the blood and producing urine. Nephrons, the functional units of the kidneys, remove excess water, electrolytes, and metabolic waste products through filtration, reabsorption, and secretion processes. These processes ensure the removal of waste while maintaining the proper balance of essential substances in the body. 4. Gaseous excretion. Gaseous excretion involves the removal of waste gases produced during metabolic processes. In organisms, such as humans, carbon dioxide, CO2, is a waste product resulting from cellular respiration. It is eliminated through the respiratory system via exhalation. Gaseous exchange occurs in the lungs, where oxygen is taken in, and carbon dioxide is released. 5. Role of the skin in excretion. The skin plays a significant role in excretion through the production and elimination of sweat. Sweat is primarily composed of water, salt, and small amounts of metabolic waste products, such as urea. Sweating helps regulate body temperature and aids in the removal of waste substances from the body's surface. This excretory function of the skin complements the roles of other excretory organs in maintaining overall homeostasis. 6. Importance of excretion. Excretion is vital for maintaining internal balance, preventing the buildup of toxic substances, and supporting overall health. By removing waste products and maintaining proper fluid and electrolyte balance, excretion helps regulate blood pressure, pH levels, and the concentrations of essential substances in the body. Efficient excretion is crucial for the optimal functioning of various physiological processes and the overall well-being of organisms. Understanding the mechanisms and importance of excretion provides insights into how organisms maintain internal balance and remove waste products to support optimal health. The coordination of excretory processes ensures the elimination of harmful substances while preserving essential nutrients and maintaining stable internal conditions. The study of excretion has broader implications in fields such as physiology, biology, and medicine. It aids in the diagnosis and treatment of renal disorders, the development of improved drug therapies, and the exploration of metabolic pathways. By gaining a comprehensive understanding of excretion, researchers and healthcare professionals can enhance their knowledge of how waste removal processes impact overall health and contribute to the prevention and management of various diseases and disorders. Conducting controlled investigations, exploring pupil size changes in response to light intensity variations, OR, DR. In this section, we will delve into the significance of conducting controlled investigations to observe changes in pupil size in response to variations in light intensity. By providing a detailed explanation, we can understand the rationale behind these investigations, the role of controlled conditions, and the insights gained regarding the pupillary response to different light levels. 1. Understanding pupillary response. The pupil is the dark circular opening in the center of the iris, and it plays a crucial role in regulating the amount of light that enters the eye. The size of the pupil changes in response to different lighting conditions, with a larger pupil allowing more light to enter and a smaller pupil restricting light entry. Investigating pupillary response helps us understand the intricate mechanisms by which the eye adjusts to different light levels. 2. Controlled investigations. Conducting controlled investigations ensures that the observed changes in pupil size can be attributed to variations in light intensity alone, minimizing the influence of other factors. By carefully controlling and manipulating the light levels while keeping other variables constant, researchers can establish a cause and effect relationship between light intensity and pupil size changes. 3. Experimental design. In controlled investigations, the experimental design is crucial. Researchers adjust the light intensity using calibrated light sources or controlled lighting conditions. They measure and record the initial pupil size under specific lighting conditions and then expose the subjects to different light intensities while monitoring the subsequent changes in pupil size. This process allows for the systematic collection of data and the identification of patterns or trends. 4. Data collection and analysis. During the investigations, researchers record the changes in pupil size in response to variations in light intensity. They may use specialized equipment, such as pupillometers or digital imaging techniques, to obtain accurate measurements. The data collected is then analyzed to determine the relationship between light intensity and pupillary response. Statistical methods and graphical representations aid in interpreting the results and drawing meaningful conclusions. 5. Interpretation of findings. The findings of controlled investigations provide insights into how the pupillary response adapts to changes in light involved and the adaptive functions of the pupillary reflex. Likewise, analyzing the structures of dissected eyes provides a deeper understanding of their roles in vision, light refraction, and the transmission of visual information to the brain. Observation and recording skills are fundamental for gaining a comprehensive understanding of pupil dynamics and the anatomical structures of mammalian eyes. By honing these skills, students develop the ability to make precise and detailed observations, record their findings systematically, and engage in critical analysis. This process enables them to unravel the intricate complexities of these organs and their significance in visual perception. The observation and recording of pupil size changes and dissected eye structures contribute to advancements in fields such as ophthalmology, neuroscience, physiology, and vision research. They aid in the diagnosis and treatment of eye disorders, the exploration of visual perception mechanisms, and the development of innovative approaches in eye care. Furthermore, these skills enhance our overall understanding of the interplay between anatomical structures, physiological processes, and vision-related phenomena. By engaging in the process of observation and recording, students develop important scientific skills, such as attention to detail, accurate documentation, and critical analysis. They cultivate a scientific mindset, curiosity, and the ability to interpret and communicate their findings effectively. These skills extend beyond the study of pupil dynamics and dissected eyes, empowering students to approach scientific inquiry with rigor and precision in various disciplines and research endeavors. Drawing, depicting and labeling mammalian eye structures, DR. In this section, we will explore the significance of drawing as a skill in accurately depicting and labeling the structures of mammalian eyes. By providing a detailed explanation, we can understand how drawing enhances our understanding of the anatomical features and functional aspects of these remarkable organs. 1. Drawing as a visual learning tool. Drawing serves as a powerful visual learning tool that allows us to create representations of anatomical structures. By engaging in the process of drawing, students can observe, analyze, and depict the intricate details of mammalian eye structures. Through drawing, they gain a deeper understanding of the spatial relationships, sizes, shapes, and relative positions of different components. 2. Accurate representation of eye structures. Drawing enables students to accurately represent the structures of mammalian eyes. They can depict the cornea, lens, iris, ciliary body, retina, optic nerve, and other associated structures with precision. Through careful observation and attention to detail, students develop the ability to depict the unique characteristics of each structure, such as their shapes, colors, textures, and anatomical connections. 3. Labeling for comprehensive understanding. Alongside drawing, labeling plays a crucial role in enhancing our understanding of mammalian eye structures. By labeling the drawn diagrams, students reinforce their knowledge and comprehension of the different components. Labels provide a visual representation of the names and functions of each structure, aiding in the retention of information and facilitating clear communication. 4. Visualization of functional aspects. Drawing allows us to visualize the functional aspects of mammalian eyes. For example, students can depict how light is refracted by the cornea and lens to form clear images on the retina. They can illustrate the role of the iris in controlling the size of the pupil and regulating the amount of light entering the eye. By drawing and labeling these functional aspects, students gain insights into how the structures work together to facilitate vision. 5. Enhancing observation and analytical skills. Engaging in drawing exercises enhances students' observation and analytical skills. Through careful observation of mammalian eye structures, they develop a keen eye for detail and the ability to identify and depict specific features accurately. Drawing encourages students to examine the structures from multiple angles and perspectives, fostering a deeper understanding of their three-dimensional nature. Drawing and labeling the structures of mammalian eyes contribute to the advancement of knowledge in fields such as anatomy, ophthalmology, biology, and medical education. Accurate drawings and labels facilitate clear and effective communication of anatomical concepts, aiding in the teaching and learning of eye anatomy. Additionally, drawing fosters artistic expression and creativity while promoting a multidimensional understanding of anatomical structures. By developing drawing skills, students strengthen their observational abilities, improve spatial reasoning, and enhance their understanding of mammalian eye structures. The ability to visually represent and comprehend these intricate structures equips students with a solid foundation for further exploration of ocular anatomy, vision science, and medical studies. Drawing not only aids in understanding anatomical features but also serves as a valuable tool for communication and collaboration among healthcare professionals, researchers, and educators. Accurate drawings and labels facilitate clear and effective communication of anatomical concepts and findings, promoting accurate diagnoses, research advancements, and educational materials. Movement, exploring the dynamics of physical motion and biological mobility. In this section, we will delve into the significance of movement as a fundamental aspect of both physical motion and biological mobility. By providing a detailed explanation, we can understand the diverse aspects of movement, ranging from locomotion in animals to the physical principles that govern motion in the physical world. 1. Overview of movement. Movement refers to the act or process of changing position, posture, or location. It is an inherent characteristic of living organisms and a fundamental principle of the physical world. Movement allows organisms to interact with their environment, respond to stimuli, seek resources, and carry out essential functions necessary for survival and growth. 2. Biological mobility. In the context of living organisms, movement encompasses biological mobility. Animals, including humans, exhibit various forms of movement, such as walking, running, flying, swimming, and crawling. These movements are achieved through the coordinated actions of muscles, joints, and nervous systems. Biological mobility plays a crucial role in hunting, escaping predators, finding mates, acquiring food, and exploring new environments. 3. Locomotion in animals. Locomotion refers to the movement of an organism from one place to another. Animals have evolved diverse mechanisms of locomotion based on their anatomical structures, physiology, and ecological adaptations. For instance, quadrupeds utilize their limbs to move on land, birds employ wings for aerial locomotion, and fish propel themselves through water using fins or body undulations. The study of animal locomotion provides insights into the adaptations and efficiencies required for different modes of movement. 4. Physical principles of motion. Movement in the physical world follows fundamental principles of physics. These principles, such as Newton's laws of motion, govern the behavior of objects and bodies in motion. Understanding these principles allows us to analyze and predict the trajectories, velocities, accelerations, and forces involved in physical movement. From the motion of celestial bodies to the mechanics of machinery and vehicles, the study of physical motion has wide-ranging applications. 5. Applications in sports and recreation. Movement has significant implications in sports, recreation, and physical activities. Athletes employ techniques, strategies, and training methods to optimize their movements and achieve peak performance. Understanding biomechanics, kinetics, and kinematics of movement helps in refining athletic techniques, preventing injuries, and enhancing overall physical fitness. 6. Motor skills and coordination. Movement involves the development and mastery of motor skills and coordination. Motor skills encompass a range of abilities, including balance, agility, flexibility, strength, and precision. Coordinated movements require the integration of sensory input, muscle control, and central nervous system processing. Enhancing motor skills and coordination enables individuals to perform tasks more efficiently, improve athletic performance, and engage in various physical activities with ease. The exploration of movement encompasses both the biological mobility of organisms and the physical principles governing motion in the physical world. Understanding the intricacies of movement provides insights into the dynamic nature of life, the mechanics of physical objects, and the potential for optimizing human performance. The study of movement has broad implications in fields such as biology, biomechanics, kinesiology, sports science, robotics, physics, and more. It contributes to advancements in healthcare, sports performance, physical rehabilitation, biomechanical engineering, and the development of assistive technologies. By unraveling the mechanisms and principles of movement, researchers and practitioners can enhance human capabilities, improve athletic performance, and advance scientific understanding. Moreover, the understanding of movement extends beyond scientific and academic realms. It promotes an active and healthy lifestyle, encourages physical fitness, and fosters an appreciation for the beauty and complexity of the natural world. Movement serves as a catalyst for exploration, self-expression, creativity, and personal development. By exploring the dynamics of movement, we gain a deeper understanding of the intricate interplay between biological mobility and physical motion. This knowledge fuels our curiosity, inspires innovation, and empowers us to appreciate and harness the power of movement in all its forms. Investigate growth in living organisms, unveiling the dynamics of development through measurements and observations, OR, DR, AI, PD. In this section, we will explore the significance of investigating growth in living organisms through simple measurements and observations. By providing a detailed explanation, we can understand the methodologies involved, the role of observation and measurement skills, as well as the significance of analysis and planning in uncovering the fascinating dynamics of development in various organisms. 1. Growth in living organisms. Growth is a fundamental aspect of living organisms and refers to the increase in size, mass, or complexity over time. Investigating growth allows us to understand the processes involved in development, from the cellular level to the organism as a whole. By studying growth, we gain insights into how living organisms mature, adapt to their environment, and reach their full potential. 2. Importance of simple measurements and observations. Simple measurements and observations are essential tools in investigating growth. By carefully recording and quantifying changes in size, weight, or other relevant parameters, we can track the progress of growth in living organisms. Additionally, detailed observations of morphological changes, behavioral patterns, and physiological adaptations provide valuable information about the stages and mechanisms of growth. 3. Observation and recording skills, ORR. Observation and recording skills are crucial in investigating growth. Students observe living organisms over time, noting any changes in appearance, behavior, or other observable characteristics. By systematically recording these observations, they create a comprehensive record of growth patterns, allowing for analysis, comparison, and the identification of developmental milestones. 4. Measurement and data collection, DR. Accurate measurement techniques play a vital role in investigating growth. Students use appropriate instruments and procedures to collect quantitative data, such as height, weight, or other measurable attributes. By employing standardized measurement methods, they ensure consistency and reliability in their findings. Precise measurements provide valuable data for tracking growth rates, identifying growth spurts, and analyzing growth patterns. 5. Analysis and interpretation, AI. Analysis and interpretation of collected data are essential for uncovering insights into growth patterns and developmental processes. Students employ statistical methods and graphical representations to analyze the data, identify trends, and draw meaningful conclusions. By interpreting the results, they gain a deeper understanding of growth dynamics, factors influencing growth rates, and potential correlations with environmental conditions or genetic factors. 6. Planning and designing investigations, PD. Planning and designing investigations are critical in studying growth in living organisms. Students develop a structured approach, outlining the objectives, variables to be measured, and the appropriate methods for data collection. By considering factors such as sample size, experimental conditions, and controls, they ensure the validity and reliability of their findings. Effective planning and design allow for a systematic exploration of growth phenomena and facilitate meaningful comparisons between different organisms or experimental conditions. Investigating growth in living organisms through simple measurements and observations contributes to various fields, including biology, ecology, agriculture, and medicine. It aids in understanding growth patterns in plants, animals, and microorganisms, informing cultivation practices, and promoting animal welfare. Additionally, investigating growth is instrumental in studying human development, identifying growth disorders, and advancing medical research. Furthermore, the investigation of growth cultivates essential scientific skills. Observation and recording skills foster attention to detail and accurate documentation, enhancing scientific literacy and critical thinking. Measurement skills promote precision and consistency, ensuring reliable data collection. Analysis and interpretation skills encourage data-driven decision-making and help unravel the underlying processes and mechanisms driving growth. By conducting investigations into growth, students gain a holistic understanding of the dynamic nature of living organisms and the intricate processes that shape their development. It sparks curiosity, encourages scientific inquiry, and fosters an appreciation for the complexity and diversity of life. Moreover, investigating growth empowers individuals to make informed decisions about cultivation practices, healthcare interventions, and sustainable development strategies. In conclusion, investigating growth in living organisms through simple measurements and observations unveils the fascinating dynamics of development. By honing observation, measurement, analysis, and planning skills, individuals can unravel the intricacies of growth, contributing to scientific knowledge and the betterment of various fields. This exploration promotes a deeper appreciation for the beauty of life's transformative processes and encourages lifelong curiosity and learning. Examine and draw, unveiling the intricacies of insects and wind-pollinated flowers, OR, DR, AI, PD. In this section, we will explore the significance of examining and drawing various parts of insects and wind-pollinated flowers. By providing a detailed explanation, we can understand the methodologies involved, the role of observation and drawing skills, as well as the significance of analysis and planning in uncovering the intricate details of these fascinating organisms. 1. Importance of examination and drawing. Examination and drawing are essential techniques for understanding the anatomy and structures of insects and wind-pollinated flowers. Through careful examination, we can observe the diverse features, adaptations, and functional aspects of these organisms. Drawing allows us to create accurate representations of their various parts, enhancing our understanding of their form and function. 2. Observation and recording skills, ORR. Observation and recording skills are fundamental in examining insects and wind-pollinated flowers. Students observe these organisms, paying close attention to their distinct features, such as body segments, appendages, sensory structures, and reproductive organs. By recording their observations systematically, they create a comprehensive visual record for analysis, comparison, and further study. 3. Drawing and labeling skills, DR. Drawing and labeling skills play a crucial role in accurately representing the various parts of insects and wind-pollinated flowers. Students create detailed and proportional drawings that depict the specific characteristics of these organisms. By adding labels to their drawings, they enhance their understanding and communication of the different parts and their functions. 4. Analysis and interpretation, AI. Analysis and interpretation of the observed and drawn features are essential for uncovering insights into the structures and adaptations of insects and wind-pollinated flowers. Students analyze the relationships between different parts, identify specialized structures, and interpret their functional significance. By examining the unique features, such as wings, mouthparts, antennae, or specialized floral structures, they gain insights into how these organisms interact with their environment. 5. Planning and designing investigations, PD. Planning and designing investigations are crucial in the examination and drawing of insects and wind-pollinated flowers. Students develop a structured approach, outlining the specific parts to focus on, selecting suitable specimens, and planning the drawing techniques to be employed. By considering factors such as magnification, lighting, and the use of reference materials, they ensure accurate and detailed representations. The examination and drawing of insects and wind-pollinated flowers contribute to various fields, including entomology, botany, ecology, and agriculture. It aids in the identification of insect species, the study of insect behavior, the understanding of plant-pollinator interactions, and the classification of plant species. Additionally, examining and drawing these organisms fosters an appreciation for the intricate adaptations and biodiversity in nature. Moreover, this practice cultivates essential scientific skills. Observation and recording skills enhance attention to detail, accuracy, and documentation, fostering scientific literacy and critical thinking. Drawing and labeling skills promote visual representation, precision, and clear communication of scientific concepts. Analysis and interpretation skills encourage the identification of patterns, the recognition of functional adaptations, and the formulation of hypotheses. By examining and drawing insects and wind-pollinated flowers, students gain a deeper understanding of the complex structures, functions, and ecological roles of these organisms. It sparks curiosity, encourages scientific inquiry, and fosters an appreciation for the interconnections between plants, animals, and the environment. Additionally, this practice cultivates artistic expression, attention to detail, and an appreciation for the beauty and diversity of the natural world. In conclusion, the examination and drawing of various parts of insects and wind-pollinated flowers unveil the intricacies of these remarkable organisms. By honing observation, drawing, analysis, and planning skills, individuals can unravel the fascinating details of their structures and adaptations, contributing to scientific knowledge and the understanding of ecological interactions. This exploration promotes a deeper appreciation for the beauty and complexity of nature and encourages lifelong curiosity and learning. Skills, nurturing proficiencies for personal and professional growth. Skills are the acquired proficiencies that enable individuals to perform tasks, solve problems, and achieve goals effectively. In this section, we will explore the significance of skills in personal and professional development, emphasizing their role in enhancing capabilities, fostering success, and promoting continuous growth. 1. Importance of skills. Skills are essential in navigating various aspects of life. They encompass a wide range of competences, including technical skills specific to a particular field and transferable skills that can be applied across different domains. Skills empower individuals to adapt to new situations, communicate effectively, collaborate with others, and accomplish desired outcomes. 2. Technical skills. Technical skills refer to the specific competencies required in a particular field or profession. These skills are obtained through formal education, training, or hands-on experience. Examples of technical skills include programming, data analysis, graphic design, financial modeling, and laboratory techniques. Technical skills are essential for professional growth and advancement within specialized domains. 3. Transferable skills. Transferable skills, also known as soft skills or general competencies, are versatile abilities that can be applied across different roles, industries, and contexts. These skills include communication, critical thinking, problem-solving, teamwork, adaptability, leadership, and emotional intelligence, among others. Transferable skills contribute to success in various professional settings and promote effective collaboration and innovation. 4. Continuous skill development. Skill development is a lifelong process that requires continuous learning and improvement. As technology advances and industries evolve, individuals need to invest in ongoing skill development to stay relevant and competitive. Continuous learning through professional development programs, training courses, certifications, and self-directed learning is crucial for personal growth and maintaining a competitive edge. 5. Skill application in various contexts. Skills are not confined to specific domains but can be applied in various contexts. For instance, effective communication skills are essential in personal relationships, team collaborations, customer interactions, and leadership roles. Problem-solving skills are valuable in addressing challenges both at work and in personal life. The ability to adapt and embrace change is crucial for navigating uncertainties and seizing opportunities in any context. 6. Skill enhancement and mastery. Skill development involves deliberate practice, feedback, and continuous improvement. By actively seeking opportunities to apply and enhance their skills, individuals can progress from novice to mastery level. Consistent practice, seeking constructive feedback, and adopting a growth mindset enable individuals to refine their abilities, overcome limitations, and continually improve their performance. Skills empower individuals to thrive in various areas of life. They enhance personal effectiveness, boost self-confidence, and contribute to overall well-being. Moreover, possessing a diverse range of skills expands opportunities, as individuals can adapt to different roles, industries, and the evolving demands of the job market. Recognizing the importance of skills and actively developing them opens doors to personal and professional growth. It equips individuals with the capabilities needed to overcome challenges, embrace new opportunities, and make meaningful contributions to their communities and organizations. By nurturing and expanding their skill set, individuals foster continuous learning, adaptability, and resilience, enabling them to navigate an ever-changing world with confidence and success. Observation and recording, unveiling the dynamics of growth through detailed observations and precise measurements, OR. In this section, we will explore the significance of observation and recording as essential skills in understanding the intricate process of growth in living organisms. By providing a detailed explanation, we can understand the methodologies involved, the role of keen observation, accurate recording, and the significance of these skills in unraveling the complexities of growth. 1. Keen observation. Keen observation is a fundamental aspect of studying growth in living organisms. Students carefully observe the organisms, paying attention to subtle changes, growth patterns, and developmental milestones. By closely observing the organisms, students can identify physical changes, such as an increase in size, the emergence of new structures, or the maturation of existing features. 2. Recording measurements. Accurate measurement and recording of data are crucial in studying growth. Students take precise measurements related to growth parameters, such as height, weight, length, or other relevant characteristics. By using appropriate tools and techniques, they ensure the reliability and consistency of their measurements. Recording these measurements in a structured manner allows for further analysis and comparison. 3. Documenting developmental stages. Through detailed observations and recordings, students document the various developmental stages of the organisms. They note the sequence of changes, identify key milestones, and record the duration of each stage. This documentation provides valuable insights into the growth trajectory, the timing of specific developmental events, and the overall pace of growth. 4. Tracking growth rates. By consistently making observations and recording measurements at regular intervals, students can track the growth rates of living organisms. They analyze the data collected over time to identify growth spurts, periods of steady growth, or potential fluctuations in growth rates. Tracking growth rates contributes to a deeper understanding of the factors influencing growth and the dynamics of development. 5. Analysis and interpretation. Analysis and interpretation of the recorded observations and measurements allow students to gain meaningful insights into the growth process. By analyzing the data, students can identify trends, patterns, and correlations related to growth. They interpret the findings, draw conclusions, and formulate hypotheses about the factors that may influence growth, such as genetics, nutrition, environment, or other external factors. Observation and recording skills are fundamental for gaining a comprehensive understanding of the growth process in living organisms. Through keen observation and accurate recording, students develop the ability to detect subtle changes, document developmental milestones, and track growth rates. These skills lay the foundation for further analysis, interpretation, and scientific inquiry. The observations and recorded measurements of growth in living organisms contribute to advancements in various fields, including biology, ecology, agriculture, and medicine. They aid in understanding growth patterns, optimizing cultivation practices, and identifying potential abnormalities or growth-related disorders. Furthermore, these skills enhance scientific literacy, critical thinking, and attention to detail, preparing students for future scientific investigations and research endeavors. By honing their observation and recording skills, students develop important scientific proficiencies that extend beyond the study of growth in living organisms. These skills foster curiosity, attention to detail, and accurate documentation, enabling students to approach scientific inquiry with rigor and precision in various disciplines and research endeavors. Moreover, these skills are transferable and applicable to other areas of life, such as data collection and analysis, problem-solving, and decision-making. In conclusion, observation and recording skills play a crucial role in studying growth in living organisms. By carefully observing and accurately recording measurements, students gain insights into the dynamics of growth, document developmental milestones, and track growth rates. These skills foster scientific inquiry, critical thinking, and attention to detail, preparing students for future scientific pursuits and equipping them with valuable proficiencies for success in various domains. Drawing, depicting and labeling the intricate parts of insects and wind-pollinated flowers, Dr. In this section, we will explore the significance of drawing as a skill in accurately representing and labeling the various parts of insects and wind-pollinated flowers. By providing a detailed explanation, we can understand how drawing enhances our understanding of the anatomical features and functional aspects of these fascinating organisms. 1. Drawing as a visual learning tool. Drawing serves as a powerful visual learning tool that allows us to create representations of the intricate parts of insects and wind-pollinated flowers. By engaging in the process of drawing, students can observe, analyze, and depict the unique characteristics of each part. Through drawing, they gain a deeper understanding of the shapes, sizes, textures, and arrangements of these vital components. 2. Accurate representation of insect parts. Drawing enables students to accurately represent the various parts of insects. They can depict the body segments, appendages, such as antennae, legs, and wings, head structures, including eyes, mouthparts, and sensory organs, and reproductive structures. By paying attention to detail and observing these features closely, students can depict the specific characteristics of each part, such as their shapes, sizes, colors, and anatomical connections. 3. Depiction of wind-pollinated flower structures. Drawing allows students to depict the structures of wind-pollinated flowers with precision. They can illustrate the key components of these flowers, such as sepals, petals, stamens, including anthers and filaments, carpels, including the stigma, style, and ovary, and other associated structures. By accurately representing these structures, students can visualize how wind facilitates the transfer of pollen for pollination. 4. Labeling for comprehensive understanding. Alongside drawing, labeling plays a crucial role in enhancing our understanding of the various parts of insects and wind-pollinated flowers. By adding labels to their drawings, students reinforce their knowledge and comprehension of each part's name and function. Labels provide a visual representation of the specific features, aiding in the retention of information and facilitating clear communication. 5. Fostering attention to detail and observation skills. Engaging in drawing exercises fosters attention to detail and sharpens observation skills. By carefully observing the intricate parts of insects and wind-pollinated flowers, students develop a keen eye for the nuances of shape, texture, and color. Drawing encourages students to examine these parts from different angles and perspectives, fostering a deeper understanding of their three-dimensional nature and variations across species. Drawing and labeling the various parts of insects and wind-pollinated flowers contribute to the advancement of knowledge in fields such as entomology, botany, ecology, and scientific illustration. Accurate drawings and labels facilitate clear and effective communication of anatomical concepts, aiding in the teaching and learning of insect and plant anatomy. Additionally, drawing fosters artistic expression and creativity while promoting a multidimensional understanding of the intricate structures and adaptations of these organisms. By developing drawing skills, students strengthen their observational abilities, improve spatial reasoning, and enhance their understanding of the diverse parts and structures of insects and wind-pollinated flowers. The ability to visually represent and comprehend these intricate components equips students with a solid foundation for further exploration of entomology, plant biology, scientific illustration, and related fields. Drawing not only aids in understanding anatomical features but also serves as a valuable tool for communication and documentation among scientists, researchers, and educators. Accurate drawings and labels facilitate clear and effective communication of anatomical concepts, aiding in research, species identification, and educational materials. In conclusion, drawing skills are crucial in accurately representing and labeling the various parts of insects and wind-pollinated flowers. By honing these skills, students enhance their observational abilities, improve their understanding of anatomical structures, and foster creativity. Drawing serves as a valuable tool for communication, documentation, and promoting a deeper appreciation of the intricacies of the natural world. Analysis and interpretation, unveiling insights from growth investigations through data analysis and findings interpretation, AI. In this section, we will explore the significance of analysis and interpretation as essential skills in understanding the data collected during growth investigations. By providing a detailed explanation, we can understand the methodologies involved, the role of data analysis, and the significance of interpretation in unraveling valuable insights related to growth. 1. Importance of analysis and interpretation. Analysis and interpretation are crucial steps in the scientific process that allow students to make sense of the data collected during growth investigations. These skills enable students to identify patterns, relationships, and trends within the data, leading to a deeper understanding of the growth process. Interpretation helps connect the observed data to meaningful explanations and broader scientific concepts. 2. Data analysis. Data analysis involves organizing, manipulating, and summarizing the collected data to identify meaningful patterns and trends. Students use various statistical and computational techniques, such as calculating averages, creating graphs, and conducting comparative analyses. Through data analysis, students gain insights into the factors influencing growth, growth rates, and potential correlations with environmental conditions or genetic factors. 3. Identifying patterns and trends. Analysis enables students to identify patterns and trends within the data collected during growth investigations. By examining the data over time or across different experimental conditions, students can recognize regularities, fluctuations, or notable deviations. Identifying patterns and trends aids in understanding growth dynamics, critical developmental stages, and the impact of various factors on the growth process. 4. Drawing conclusions. Through interpretation, students draw meaningful conclusions based on the analyzed data. They synthesize their findings, considering the patterns, trends, and relationships identified. Interpretation involves connecting the data to underlying scientific principles, theories, or hypotheses. By drawing conclusions, students derive insights and make informed statements about the growth process, factors influencing growth, and potential implications. 5. Relating findings to scientific concepts. Interpretation allows students to relate their findings to established scientific concepts and theories. By integrating their observations, data analysis, and conclusions, students can connect their findings to existing knowledge in the field of growth and development. This process enriches their understanding of the broader scientific context, contributing to the advancement of scientific knowledge. 6. Communication of findings. Effective communication of findings is a crucial aspect of analysis and interpretation. Students should be able to articulate their observations, data analysis methods, and conclusions in a clear and concise manner. By effectively communicating their findings, students facilitate knowledge sharing, collaboration, and peer feedback, contributing to the collective understanding of growth processes. Analysis and interpretation of data collected during growth investigations provide valuable insights into the intricacies of the growth process. These skills foster critical thinking, scientific reasoning, and the ability to draw evidence-based conclusions. Moreover, they encourage students to think beyond the collected data and consider the broader implications of their findings. The application of analysis and interpretation extends beyond growth investigations. These skills are fundamental to scientific research, data-driven decision-making, and problem-solving in various disciplines. Developing proficiency in analysis and interpretation equips students with transferable skills that can be applied across scientific, academic, and professional domains. In conclusion, analysis and interpretation are vital skills for understanding the data collected during growth investigations. By analyzing the data, identifying patterns and trends, and drawing conclusions, students gain valuable insights into the growth process. These skills foster critical thinking, scientific reasoning, and effective communication of findings. By applying analysis and interpretation skills, students contribute to the advancement of scientific knowledge and develop important proficiencies applicable in diverse contexts. Planning and designing. Crafting well-structured investigations for comprehensive exploration of growth, PD. In this section, we will explore the significance of planning and designing as essential skills for conducting investigations related to growth. By providing a detailed explanation, we can understand the methodologies involved, the role of planning and designing, and the significance of these skills in ensuring a comprehensive exploration of growth phenomena. 1. Importance of planning and designing. Planning and designing investigations are critical steps in the scientific process as they provide a structured framework for conducting studies related to growth. These skills allow students to define clear objectives, identify variables to be measured and controlled, select appropriate methods, and determine the necessary resources for their investigations. Through careful planning and design, students can ensure the validity, reliability, and effectiveness of their studies. 2. Defining clear objectives. Planning involves defining clear objectives that outline the specific goals and expected outcomes of the investigation. Students establish what they intend to achieve and the questions they seek to answer. Clear objectives provide a direction for the study and guide subsequent steps, ensuring a focused and purposeful exploration of growth-related phenomena. 3. Identifying variables. Designing investigations involves identifying the variables that will be measured, manipulated, or controlled during the study. Variables may include factors that influence growth, such as environmental conditions, genetic traits, nutrient availability, or other relevant parameters. By identifying and controlling variables, students can isolate and examine their individual effects on the growth process. 4. Selecting appropriate methods. Planning and designing require students to select appropriate methods for data collection, measurement, and analysis. This involves determining the most suitable techniques, tools, and instruments to be used in the investigation. Students consider factors such as accuracy, precision, practicality, and ethical considerations when selecting methods. By employing appropriate methods, they ensure reliable and meaningful results. 5. Resource allocation. Planning involves identifying and allocating the necessary resources for the investigation. This includes considering the availability of materials, equipment, time, and human resources required to conduct the study. Adequate resource allocation ensures that students have the necessary means to carry out their investigations effectively and efficiently. 6. Consideration of controls and variables. Planning and designing investigations necessitate the consideration of controls and variables. Students identify control groups or conditions that provide a baseline for comparison and ensure that observed changes are attributed to the variables being studied. By controlling extraneous variables, students can isolate the effects of specific factors on growth and obtain more accurate and reliable results. Planning and designing investigations related to growth provide a structured approach to exploring complex phenomena. These skills foster critical thinking, hypothesis formation, and the ability to design controlled experiments. Moreover, they cultivate attention to detail, organization, and effective time management, all of which contribute to successful scientific inquiry. Effective planning and designing extend beyond growth investigations and are transferable skills applicable in various scientific, academic, and professional settings. The ability to plan and design investigations is valuable in fields such as biology, ecology, agriculture, medicine, and many others that involve systematic inquiry and data-driven decision-making. In conclusion, planning and designing are crucial skills for conducting investigations related to growth. By defining clear objectives, identifying variables, selecting appropriate methods, and allocating resources, students ensure a well-structured and focused exploration of growth phenomena. These skills foster critical thinking, hypothesis formation, and effective scientific inquiry. By honing their planning and designing skills, students cultivate proficiencies applicable in various scientific disciplines and beyond, contributing to their overall scientific literacy and success. Irritability, sensing and responding to stimuli in living organisms. In this section, we will delve into the concept of irritability, which refers to the ability of living organisms to sense and respond to various external and internal stimuli. By providing a detailed explanation, we can understand the underlying mechanisms, the role of irritability in survival and adaptation, and its significance in the functioning of living organisms. Irritability, also known as responsiveness or excitability, is a fundamental characteristic exhibited by living organisms across different levels of complexity, from single-celled organisms to multicellular organisms. It enables organisms to perceive changes in their environment and initiate appropriate responses to ensure their survival, growth, and reproduction. 1. Sensing stimuli. Living organisms possess specialized sensory structures and mechanisms that allow them to detect a wide range of stimuli from their environment. These stimuli can be physical, chemical, or biological in nature. Examples include light, sound, temperature, touch, chemicals, hormones, and the presence of other organisms. Sensory receptors, such as photoreceptors, mechanoreceptors, chemoreceptors, and thermoreceptors, enable organisms to perceive and process these stimuli. 2. Transmission of signals. Upon sensing a stimulus, living organisms generate and transmit signals through various signaling pathways. These pathways involve the conversion of the sensory input into electrochemical signals that travel through specialized cells, such as neurons or other conducting tissues. The signals are then transmitted to specific target cells or organs that initiate the appropriate response. 3. Response to stimuli. The response to stimuli is a crucial aspect of irritability. It involves the activation of effectors, which are specialized cells, tissues, or organs responsible for carrying out the response. The response can be immediate or delayed, depending on the nature of the stimulus and the organism's physiological and behavioral capabilities. Common responses include movement, secretion, changes in metabolic activity, growth, and modifications in behavior. 4. Adaptation and survival. Irritability plays a vital role in the adaptation and survival of living organisms. It enables them to respond to changes in their environment, avoid potential dangers, seek favorable conditions, and exploit available resources. Through irritability, organisms can adjust their physiological processes, alter their behavior, and adapt to new challenges or opportunities. 5. Regulation and homeostasis. Irritability is closely linked to the regulation and maintenance of internal conditions, a process known as homeostasis. Organisms continuously monitor and respond to internal stimuli, such as changes in nutrient levels, pH, osmolarity, and hormone concentrations. By detecting and responding to these internal cues, organisms can regulate their physiological processes and maintain stable internal environments necessary for proper functioning. 6. Evolutionary significance. Irritability has significant evolutionary implications. It allows organisms to respond to changes in their environment, promoting survival, reproduction, and the transmission of favorable traits to subsequent generations. Through natural selection, organisms that exhibit efficient and adaptive responses to stimuli have a greater likelihood of thriving in their respective habitats, contributing to the overall diversity and success of life on Earth. The study of irritability encompasses various scientific disciplines, including physiology, neuroscience, ethology, and ecology. It helps us understand the intricate mechanisms by which organisms sense and respond to stimuli, unraveling the complexities of their behavior, adaptations, and interactions with their environment. Irritability is not limited to complex organisms but is also exhibited by simpler life forms. Single-celled organisms, such as bacteria, display irritability by responding to changes in nutrient availability, temperature, light, and other environmental factors. This demonstrates the pervasive nature of irritability across the spectrum of life. In conclusion, irritability is a fundamental characteristic of living organisms that enables them to sense and respond to stimuli. Through specialized sensory mechanisms, signal transmission, and appropriate responses, organisms can adapt, survive, and maintain homeostasis. Understanding irritability provides valuable insights into the complex and dynamic nature of life, highlighting the intricate interplay between organisms and their environment. Observe and analyze the responses of plants to stimuli, unveiling the complex interactions of light, touch, and gravity, OR, DR, AI, PD. In this section, we will explore the significance of observing and analyzing the responses of plants to stimuli such as light, touch, and gravity. By providing a detailed explanation, we can understand the methodologies involved, the role of observation, and the significance of these skills in unraveling the complex interactions between plants and their environment. 1. Observation and recording, OR. Observing the responses of plants to stimuli is a fundamental step in understanding their behavior and adaptive mechanisms. Students carefully observe and record the changes and reactions that occur when plants are exposed to stimuli. These observations may include changes in growth patterns, movement of plant parts, alterations in pigmentation, or modifications in the orientation of plant organs. 2. Drawing, DR. Drawing plays a crucial role in accurately representing and documenting the observed responses of plants. Students use their drawing skills to depict the changes in plant morphology, the position and movement of plant parts, and any other noticeable alterations resulting from the stimuli. By accurately depicting the observed responses, students enhance their understanding of the underlying mechanisms and facilitate clear communication of their findings. 3. Analysis and interpretation, AI. Analyzing the observed data allows students to identify patterns, trends, and correlations in the responses of plants to stimuli. Through data analysis, students can uncover the specific effects of light, touch, and gravity on plant growth, development, and behavior. Interpretation involves connecting the observed responses to established scientific concepts, theories, and hypotheses, deepening our understanding of plant physiology and ecology. 4. Planning and designing, PD. Planning and designing investigations related to plant responses to stimuli are essential to ensure the validity and reliability of the results. Students carefully plan and design controlled experiments, considering variables such as light intensity, duration of exposure, intensity of touch, or angle of gravity. Proper planning and design allow students to isolate and manipulate specific factors, providing a comprehensive understanding of how plants respond to different stimuli. Observing and analyzing the responses of plants to stimuli contribute to advancements in various fields, including plant biology, agriculture, ecology, and environmental science. It enables us to understand the adaptive strategies of plants, their responses to environmental cues, and their interactions with other organisms. Furthermore, these skills foster scientific inquiry, critical thinking, and attention to detail, preparing students for future scientific investigations and research endeavors. By honing their observation and recording skills, students develop keen eyes for detecting subtle changes in plant responses. Drawing skills enhance their ability to visually represent and communicate their observations accurately. Analysis and interpretation skills allow students to derive meaningful insights from the observed data, contributing to the broader understanding of plant physiology and ecological processes. The observation and analysis of plant responses to stimuli are not only scientifically valuable but also have practical applications. Understanding how plants respond to light, touch, and gravity aids in optimizing cultivation practices, improving crop yield, and designing sustainable agricultural systems. It also enhances our appreciation for the diversity and resilience of plant life, promoting environmental stewardship and conservation efforts. In conclusion, observing and analyzing the responses of plants to stimuli such as light, touch, and gravity are essential for understanding their behavior and adaptive mechanisms. By honing their observation, drawing, analysis, and interpretation skills, students gain insights into the intricate interactions between plants and their environment. These skills foster scientific inquiry, critical thinking, and a deeper appreciation for the complexity and importance of plants in our ecosystem. Investigate and unravel the response of invertebrates to variations in light intensity, temperature, and moisture, unveiling the complexities of environmental adaptations, OR, DR, AI, PD. In this section, we will explore the significance of investigating and understanding the response of invertebrates to variations in light intensity, temperature, and moisture. By providing a detailed explanation, we can comprehend the methodologies involved, the role of investigation, and the significance of these skills in unraveling the intricate interactions between invertebrates and their environment. Observation and recording, OR. Observing the responses of invertebrates to variations in light intensity, temperature, and moisture is a fundamental step in studying their behavior and physiological adaptations. Students carefully observe and record the changes in behavior, movement patterns, or physiological responses exhibited by invertebrates under different environmental conditions. These observations provide valuable insights into how these organisms perceive and react to their surroundings. Drawing, DR. Drawing plays a crucial role in accurately representing and documenting the observed responses of invertebrates. Students utilize their drawing skills to depict the behavioral or physiological changes in invertebrates resulting from variations in light intensity, temperature, and moisture. Accurate and detailed drawings enhance our understanding of the observed responses and aid in clear communication of findings. Analysis and interpretation, AI. Analyzing the observed data enables students to identify patterns, trends, and correlations in the responses of invertebrates to environmental variations. Through data analysis, students can unravel the specific effects of light intensity, temperature, and moisture on the behavior, physiology, or life cycle of invertebrates. Interpretation involves connecting the observed responses to established scientific concepts, theories, and ecological principles, contributing to our understanding of invertebrate biology and their ecological roles. Planning and designing, PD. Planning and designing investigations related to invertebrate responses to environmental variations are essential to ensure the validity and reliability of the results. Students carefully plan and design controlled experiments, considering factors such as light intensity, temperature gradients, or moisture levels. Proper planning and design allow students to manipulate and control specific variables, providing a comprehensive understanding of how invertebrates respond to different environmental conditions. Investigating the response of invertebrates to variations in light intensity, temperature, and moisture contributes to advancements in various fields, including ecology, entomology, marine biology, and environmental science. It allows us to understand how invertebrates adapt to different environmental conditions, survive, and fulfill their ecological roles. Furthermore, these skills foster scientific inquiry, critical thinking, and attention to detail, preparing students for future scientific investigations and research endeavors. By honing their observation and recording skills, students develop keen eyes for detecting subtle behavioral or physiological changes in invertebrates. Drawing skills enhance their ability to visually represent and communicate their observations accurately. Analysis and interpretation skills allow students to derive meaningful insights from the observed data, contributing to our broader understanding of invertebrate biology and ecological processes. The investigation of invertebrate responses to variations in light intensity, temperature, and moisture has practical implications as well. Understanding how invertebrates adapt to changing environmental conditions aids in conservation efforts, pest management strategies, and ecosystem monitoring. It also enhances our appreciation for the diversity and resilience of invertebrate life and highlights their ecological significance. In conclusion, investigating and unraveling the response of invertebrates to variations in light intensity, temperature, and moisture are essential for understanding their behavior and physiological adaptations. By honing their observation, drawing, analysis, and interpretation skills, students gain insights into the complex interactions between invertebrates and their environment. These skills foster scientific inquiry, critical thinking, and a deeper appreciation for the importance of invertebrates in ecological systems. Skills, essential abilities for effective scientific inquiry and exploration. In this section, we will delve into the significance of skills in conducting effective scientific inquiry and exploration. Skills are the foundation that empowers students to engage in scientific practices, analyze data, communicate findings, and make informed conclusions. By providing a detailed explanation, we can understand the key skills required for successful scientific endeavors and their importance in advancing knowledge. 1. Observation and recording. The skill of observation and recording is fundamental to scientific inquiry. It involves keenly observing phenomena, collecting accurate and detailed data, and recording observations systematically. This skill allows students to capture and document important information that forms the basis for analysis and interpretation. 2. Drawing and illustration. Drawing and illustration skills enable students to visually represent scientific concepts, data, and observations. Through drawings, diagrams, or visual models, students can communicate complex ideas, structures, and processes effectively. These visual representations facilitate comprehension, enhance communication, and promote a deeper understanding of scientific phenomena. 3. Analysis and interpretation. Analysis and interpretation skills are critical for making sense of collected data and deriving meaningful insights. Students use these skills to identify patterns, trends, relationships, and anomalies within the data. Through analysis and interpretation, students can draw conclusions, make evidence-based claims, and contribute to the broader scientific understanding. 4. Planning and designing. The skill of planning and designing is essential for conducting well-structured scientific investigations. Students need to develop a clear plan that outlines objectives, identifies variables, determines appropriate methods, and allocates necessary resources. Effective planning and designing ensure the validity, reliability, and efficiency of scientific experiments or studies. 5. Critical thinking and problem solving. Critical thinking and problem-solving skills enable students to analyze complex situations, evaluate evidence, and develop logical solutions. These skills foster the ability to ask meaningful questions, think critically about scientific concepts, and make informed decisions based on available data. Critical thinking empowers students to approach scientific challenges with curiosity, creativity, and a systematic mindset. 6. Communication and presentation. Effective communication and presentation skills are vital for sharing scientific findings, ideas, and discoveries. Students need to convey their thoughts, data, and conclusions through oral and written communication. By developing clear and concise communication skills, students can effectively disseminate scientific knowledge, collaborate with peers, and engage with the broader scientific community. These skills are interconnected and mutually reinforcing. By honing these skills, students become better equipped scientists who can observe the natural world, conduct meaningful investigations, analyze data, draw accurate representations, make logical interpretations, and effectively communicate their findings. These skills are not only valuable in scientific pursuits but also transferable to various academic and professional contexts. Developing and refining these skills fosters scientific literacy, critical thinking, and a lifelong appreciation for inquiry-based learning. Moreover, these skills enable students to contribute to scientific advancements, address real-world challenges, and engage in evidence-based decision-making. In conclusion, skills form the foundation of effective scientific inquiry and exploration. Observation and recording, drawing and illustration, analysis and interpretation, planning and designing, critical thinking and problem-solving, as well as communication and presentation skills, collectively empower students to engage in scientific practices and make meaningful contributions to the scientific community. By nurturing these skills, students become well-rounded individuals capable of understanding, analyzing, and communicating scientific concepts, paving the way for future discoveries and innovations. Observation and recording, or unveiling the intricacies of plant and invertebrate responses to stimuli through systematic observation and recording. Observation and recording, unveiling the intricacies of plant and invertebrate responses to stimuli. In this section, we will explore the significance of observation and recording as essential skills in studying the responses of plants and invertebrates to various stimuli. Through careful observation and systematic recording, we can uncover the complex behaviors, adaptations, and physiological changes exhibited by these organisms when exposed to different stimuli. Observation and recording involve keenly observing the behaviors, movements, and physiological responses of plants and invertebrates in the presence of various stimuli. By closely monitoring their reactions and documenting the observed changes, students can gain valuable insights into how these organisms perceive and interact with their environment. 1. Detailed and systematic observation. Students engage in detailed and systematic observation, paying attention to subtle changes, variations, and patterns in the behavior and responses of plants and invertebrates. They observe factors such as changes in growth patterns, movement, orientation, feeding behaviors, or physiological modifications. Detailed observation enables students to capture a comprehensive picture of the organism's responses to different stimuli. 2. Accurate recording. Accurate recording is an essential component of observation and ensures that the observed data is captured and documented faithfully. Students record their observations in a systematic manner, including the time, date, conditions, and any other relevant factors that might influence the responses of plants and invertebrates. Accurate recording allows for later analysis, interpretation, and comparison of the collected data. 3. Recognition of patterns and trends. Through consistent observation and recording, students can recognize patterns, trends, and variations in the responses of plants and invertebrates to different stimuli. By identifying recurring behaviors or physiological changes, students gain insights into the underlying mechanisms and adaptations that enable these organisms to respond to specific stimuli. Recognition of patterns and trends enhances our understanding of the complexities of these organisms' responses. 4. Documentation for further analysis. The recorded data serves as a valuable resource for further analysis and scientific inquiry. It allows students to examine the collected information, identify relationships between stimuli and responses, and draw evidence-based conclusions. By analyzing the recorded data, students can derive insights, test hypotheses, and contribute to the existing scientific knowledge in the field. Observation and recording skills are foundational to the scientific process and contribute to the advancement of various scientific disciplines, including ecology, ethology, plant physiology, and invertebrate biology. These skills foster attention to detail, precision, and scientific rigor, providing students with a solid grounding in scientific inquiry. By honing their observation and recording skills, students develop a keen eye for detecting subtle changes, behaviors, and adaptations exhibited by plants and invertebrates. These skills also enhance students' ability to effectively communicate their findings, share scientific knowledge, and engage in collaborative discussions with peers and experts. In conclusion, observation and recording are crucial skills for studying the responses of plants and invertebrates to different stimuli. Through systematic observation and accurate recording, students gain insights into the complexities of these organisms' behaviors, adaptations, and physiological changes. These skills foster scientific curiosity, attention to detail, and critical thinking, enabling students to contribute to the broader understanding of the natural world. Drawing, depicting and labeling the intricate structures of plants and invertebrates for enhanced understanding and communication. Drawing, depicting and labeling the intricate structures of plants and invertebrates. In this section, we will explore the significance of drawing as a skill in accurately representing and labeling the intricate structures of plants and invertebrates. Through detailed and precise drawings, students can enhance their understanding of these organisms' anatomy and communicate their findings effectively. Drawing plays a crucial role in scientific inquiry, particularly when it comes to representing the structures of plants and invertebrates. By honing their drawing skills, students can depict and label the intricate details of various plant and invertebrate structures, including organs, tissues, appendages, and other anatomical features. 1. Accurate representation. Drawing enables students to visually represent the structures of plants and invertebrates with accuracy and precision. By carefully observing and studying these organisms, students can translate their observations into detailed drawings that capture the specific shapes, sizes, textures, and arrangements of different structures. Accurate representation aids in developing a comprehensive understanding of these organisms' anatomical characteristics. 2. Labeling. Labeling is an integral part of drawing, as it allows students to identify and provide descriptive labels for the various structures depicted in their drawings. By labeling structures such as leaves, stems, roots, antennae, legs, or specialized organs, students enhance their understanding of the specific functions and roles these structures play in the organism's physiology, behavior, or ecological interactions. Proper labeling facilitates clear communication of the observed structures and their significance. 3. Visualization and conceptualization. Drawing supports the visualization and conceptualization of complex structures and their relationships within the context of plants and invertebrates. Through the process of creating accurate drawings, students deepen their understanding of the three-dimensional nature of these organisms' anatomy and the spatial relationships between different structures. Visualization and conceptualization foster a holistic perspective, aiding in comprehension and analysis of the organism's biological and ecological characteristics. 4. Communication of findings. Drawings serve as valuable tools for communicating scientific findings and observations. Students can use their drawings to present their research, share their understanding of plant and invertebrate structures with peers and teachers, and contribute to scientific discussions. Clear and well-labeled drawings enable others to grasp the details and nuances of the structures being depicted, facilitating effective communication and collaboration. Drawing as a skill extends beyond scientific investigations and has practical applications in various fields, including biology, zoology, botany, entomology, and scientific illustration. It fosters creativity, attention to detail, and spatial reasoning, all of which are valuable in scientific pursuits and artistic endeavors. By honing their drawing skills, students develop the ability to accurately represent and label the structures of plants and invertebrates. This skill enhances their overall scientific literacy, enabling them to analyze, interpret, and communicate scientific concepts more effectively. Furthermore, drawing promotes visual thinking, artistic expression, and a deeper appreciation for the intricacies of the natural world. In conclusion, drawing is a valuable skill for accurately representing and labeling the structures of plants and invertebrates. Through precise and detailed drawings, students deepen their understanding of these organisms' anatomy and facilitate effective communication of their observations. Developing drawing skills enhances scientific literacy, fosters creativity, and strengthens the connection between scientific inquiry and artistic expression. Analysis and interpretation, AI unveiling insights from data and interpreting organisms' responses to stimuli. Analysis and interpretation, unveiling insights from data and interpreting organisms' responses to stimuli. In this section, we will explore the significance of analysis and interpretation as crucial skills in scientific investigations. Students will learn how to analyze the data collected during investigations and derive meaningful insights about the responses of organisms to stimuli. Through thoughtful interpretation, they can deepen their understanding of the underlying mechanisms and broader implications of these responses. 1. Data analysis. Students engage in data analysis to uncover patterns, trends, and relationships within the collected data. They employ various statistical and analytical techniques to examine the data systematically. By organizing, summarizing, and visualizing the data, students can identify important features, detect significant changes, and quantify the observed responses of organisms to different stimuli. Data analysis allows for objective examination of the information collected during the investigations. 2. Deriving insights. Through data analysis, students derive insights that lead to a deeper understanding of the responses of organisms to stimuli. They look for patterns or trends that emerge from the data, enabling them to draw evidence-based conclusions and make connections between the stimuli and the observed responses. These insights contribute to a more comprehensive understanding of the organism's behavior, adaptations, or physiological changes in the context of the stimuli under investigation. 3. Exploring relationships and correlations. Analysis and interpretation involve exploring relationships and correlations between different variables. Students examine how variations in stimuli relate to the observed responses of organisms. They may identify cause and effect relationships, dependencies, or interactions between stimuli and the organism's behaviors or physiological changes. Exploring these relationships provides valuable insights into the mechanisms by which organisms perceive and respond to specific stimuli. 4. Contextual interpretation. Interpretation involves placing the observed responses of organisms within a broader context. Students consider existing scientific knowledge, theories, or ecological principles to interpret the significance and implications of the observed responses. They explore how the responses contribute to the organism's survival, adaptation, or ecological interactions. Contextual interpretation allows students to connect their findings to the broader scientific understanding of the organism's biology and ecology. Data analysis and interpretation skills are fundamental to scientific inquiry and contribute to advancements in various fields, including biology, ecology, physiology, and environmental science. These skills foster critical thinking, analytical reasoning, and evidence-based decision-making. By honing their data analysis and interpretation skills, students develop the ability to draw meaningful conclusions, make evidence-based claims, and contribute to the scientific discourse. They learn to critically evaluate and contextualize their findings, consider alternative explanations, and recognize the limitations and uncertainties in their data. These skills empower students to engage in scientific reasoning and promote a deeper understanding of the natural world. In conclusion, analysis and interpretation are essential skills for uncovering insights from data and interpreting the responses of organisms to stimuli. Through data analysis, students identify patterns and trends, while interpretation provides a contextual understanding of the observed responses. Developing these skills enhances scientific literacy, critical thinking, and the ability to contribute to scientific knowledge. By engaging in analysis and interpretation, students become active participants in the scientific process and gain a deeper appreciation for the complexities of the organism's responses to stimuli. Planning and designing, PD crafting thoughtful investigations for exploring plant and invertebrate responses to stimuli. Planning and designing, crafting thoughtful investigations for exploring plant and invertebrate responses to stimuli. In this section, we will explore the significance of planning and designing as an essential skill for conducting investigations related to plant and invertebrate responses to stimuli. Students will learn how to develop well-structured and comprehensive investigation plans that ensure the validity, reliability, and effectiveness of their scientific studies. Planning and designing investigations involve careful consideration of various factors and components to create a framework that guides the research process. This skill is crucial in developing investigations that address specific research questions, manipulate relevant variables, and yield meaningful results. 1. Defining objectives. Students begin by defining clear and specific objectives for their investigations. They identify the research questions they seek to answer and articulate the specific goals they aim to achieve. Defining objectives provides a focused direction for the investigation and ensures that the study addresses relevant aspects of plant and invertebrate responses to stimuli. 2. Identifying variables. Students identify the key variables that will be manipulated and measured in their investigations. These variables may include different types or intensities of stimuli, environmental conditions, or specific aspects of plant and invertebrate behavior or physiology. By identifying and controlling variables, students can isolate the effects of specific factors on the observed responses. 3. Selecting appropriate methods and techniques. Planning and designing involves selecting appropriate methods and techniques that align with the research objectives and variables. Students consider the most effective and reliable approaches to collect data, such as field observations, controlled experiments, or sampling techniques. The selection of methods and techniques ensures that the investigations are conducted in a systematic and rigorous manner. 4. Allocating resources. Students consider the resources required for their investigations, including equipment, materials, time, and human resources. They plan and allocate these resources efficiently to ensure that the investigations can be carried out effectively. Adequate resource allocation contributes to the smooth execution of the investigations and enhances the reliability and validity of the results. 5. Addressing ethical considerations. Planning and designing investigations also involve addressing ethical considerations related to the treatment of plants and invertebrates. Students consider the well-being and ethical treatment of organisms during the experiments or studies. They adhere to ethical guidelines and protocols, ensuring that the investigations are conducted responsibly and with respect for the welfare of the organisms involved. Effective planning and designing facilitate rigorous and systematic scientific inquiry, ensuring that the investigations are well-structured, controlled, and capable of producing reliable and meaningful results. Students develop skills in critical thinking, problem-solving, and experimental design, which are valuable across various scientific disciplines. By honing their planning and designing skills, students develop the ability to create investigation plans that optimize the potential for scientific discovery. They learn to think critically about research objectives, consider appropriate variables and methods, and anticipate potential challenges or limitations. These skills empower students to engage in scientific inquiry and contribute to the advancement of knowledge in the field of plant and invertebrate responses to stimuli. In conclusion, planning and designing investigations are crucial skills for exploring plant and invertebrate responses to stimuli. By defining objectives, identifying variables, selecting appropriate methods, allocating resources, and addressing ethical considerations, students create a strong foundation for conducting scientifically rigorous investigations. Developing these skills enhances scientific literacy, critical thinking, and the ability to contribute to scientific knowledge. Through thoughtful planning and design, students embark on a journey of scientific exploration and gain a deeper understanding of the complexities of plant and invertebrate responses to stimuli.